Can I, Andrew? Yeah, I think what Dr. Prashant is saying is probably related to that we should have a kind of visualization that whether 1 to 2, and if more are available, why not more teeth? Yes. Why to just keep limited to 3 teeth? There was another issue which one of the participants here sitting next to me also raised, which we often feel while doing an autopsy, and with this new upcoming thought on ethics of handling the dead body, the balance between when is the sample less, it shouldn't be less, and why to send more. So you're trying to strike a balance. Why to send more, and it shouldn't fall less. Now, if you are talking of long bone, it means there is a process to remove it, but at the end of it, it also mutilates the body. It also kind of makes it deformed for the attendants to take back the dead body, to cremate it or to bury it. It is at that time while we are doing an autopsy where the attendants are outside and we are saying hum ye sab nikal rahe hain. We are collecting all this and sending it for investigation, and we are trying to explain this is the case and this is why it is required. So for the long bone like femur, we normally try to do, let's take it out from the lower leg where there are two bones, tibia and fibula, both long bones. One is thinner one is relatively thicker one. So we normally take out one of these bones, and fibula, because it's thinner, it could probably make the leg look sturdier, and this would easily, because you can do a vertical cut and then put it at a back side or something. And the tibia is exposed more on the skin, with less of muscle tissue to separate it from, so it will be less messy, less time consuming. So those kind of thoughts or those kind of processes.
If you remember, he has shown a slide. If you have it is always better to have tissue. You can have thigh muscles. No need of taking bone. If thigh muscle is there, take muscle. No problem. It will not deform. Okay. But अब अगर हम ये जो हम एक लिस्ट से नीचे आते हैं ना कि ये चाहिए फिर ये चाहिए फिर ये चाहिए, this never means that we need all the samples. If you have अगर आप पूरी बॉडी है और हम सलाइवा का स्वैब ले लेते हैं, when that is good enough to prove the identity. This any stain on body is okay, tissue is okay, teeth is okay. जिसमें भी अगर if you have ample choice, बॉडी कितना डिटेरियोरेटेड है उसके हिसाब से ये लिस्ट बनती है. अब देयर इज ए स्टैंडर्ड गाइडलाइन. वो स्टैंडर्ड गाइडलाइन जिसने भी बनाई है अलग अलग तरीके की है कि बॉडी किस शेप में हो तो क्या सैंपल लिया जाए. अगर होल बॉडी इज देयर तो इट इज ऑलवेज बेटर टू हैव टिश्यू. ओके. अब बॉडी में कितना पुट्रीफैक्शन हुआ है उसके हिसाब से सैंपल धीरे धीरे नीचे आता जाएगा. ओके. इस तरीके की बात है. और किसी बोन में प्रोफाइल नहीं आएगी ऐसा नहीं है. जैसे मैं कह रहा हूँ आपसे किसी दांत से प्रोफाइल नहीं आती, ऐसा भी नहीं है. हर किसी सैंपल से जिसमें भी न्यूक्लियर डीएनए है उससे डीएनए प्रोफाइल आ जाएगी. बस फर्क ये होता है कि किसी में इतना ज्यादा डीएनए है, किसी में इतना कम है, बस. लेकिन आज की जो नीड है, ये जो गाइडलाइंस बनी थी, दिस गाइडलाइन, ऑल द गाइडलाइंस आर ओल्ड. आप मैक्सिमम लोगों के प्रेजेंटेशन में सुनोगे कि टेक 5 और 10 ml ब्लड सैंपल. मैक्सिमम प्रेजेंटेशन में आप देखोगे, मैक्सिमम बुक्स में आप देखोगे. बट व्हाट इज सेड that blood is 1 to 2 ml. अब आज की जो गाइडलाइन है, if you talk about us who are practicing DNA, we need 1 microliter of DNA. If you give referral sample, we need only 1 microliter of blood sample. Okay, so इसमें ये नहीं है कि 10 ml से ज्यादा DNA आएगा और 1 microliter से नहीं आ पाएगा. हमें जनरेट तो प्रोफाइल ही करनी है. We need DNA in picograms. So it can be generated. So if you have a choice of sample, so whatever sample is easy, you can have it.
One more thought which guides us is that if this sample is going to take long to reach FSL for you to analyze, and so we must send the bone.
Yes.
अब नहीं होता. If you talk about MP, so we have a guideline that within 15 days a case is to be sent to the laboratory. So 15 days में कुछ नहीं होता. If you don't even preserve your sample, even then 15 days में कुछ नहीं होगा. सलाइन में होता तो वो उसमें कुछ भी नहीं होता. कोई दिक्कत नहीं है उसमें.
One more. My question is to Dr. Rao and Dr. Pankaj. There was in the recent conference at Bhopal, one of the very senior scientists of probably CDFD was talking that the success rate of DNA extraction from femur is as good as 95 to 98%, and those from the small or thin bones like fibula or the radius and ulna is about to 50 or 60%. How is that correct?
Depends on the expertise of the expert. See, what I can say to all your queries is, it's all the man behind the machine which matters. Today we are going into machinization, automation and all that is causing all these problems. You have skeletal remains, you send one piece of bone. It's sufficient to come to a conclusion. Now, sir was saying some expert came and talked, it might be his lab experience, but not necessary. All bones will give 95 to 98% conclusive DNA will come out of it. A well preserved sample is always from dry bone. Any sample which is in a dried condition gives the best because DNA stops degradation after the liquid part of it gets dried up. So another gentleman was talking about the teeth. Not only roots, even the dental cavity, if you can reach to the, DNA experts can reach to the dental cavity and scrape it out, that is also sufficient to arrive at a conclusion. Because how much we need, 1 or 2 nanogram of DNA starting material to do the analysis. Earlier days it was 10 ml of blood or 15 microgram of DNA. Now sensitivity has increased. So your experience, sir, it is maybe the person who is doing, he is very bad at small bones and good at big bones.
Fault of the scientist.
It's man who is wrong.
And one more thing, one should preserve as far as possible a healthy tooth, not the decayed one.
You have no choice in sampling, sir. As it comes before us, we have to do it.
So I would like, please, let's carry on this discussion with over tea or over our hands-on things. We can take up this discussion, carry on it, because we are running short of time, and I would really like to thank Dr. Ramkishan Kumawat, who has so beautifully explained how to preserve the samples. And I think most of our doubts, most of our practical issues have and queries have been put to rest by his presentation. And now I would like to proceed for the hands-on workshop, and how it is to be done will be explained by Dr. Anuradha.
Excuse me, one thing more regarding the.
So here we are going to demonstrate the extraction of our resource persons are going to demonstrate the extraction of various biological samples from various surfaces. Here we have samples of blood, saliva, semen, body tissue, vaginal swab, bone and teeth in powdered form, and these are kept on tiles, clothing and gauze. So we have porous and non-porous both surfaces. We are going to understand how to extract the sample and how to analyze it from the resource persons. So for this we have 5 stations. So I request you all to divide yourselves among we have given you registration number from 001 to so on. So I request you all, registration number 1 to 6 will be group 1, 7 to 12 group 2 and so on. Like 13 to 18 will be group 3, 19 to 24 group 4 and 25 to 31 group 5. I think I am clear about this. And the people of AIIMS, PGs, SRs and the JRs, you please divide yourselves like 1 in each group, so that at least we have 8 people in one group, 8 or 7 people. So I request you all to please gather in the museum. There we have all the stations, and let's meet up there.
Are the samples there on each table? Okay.
We have different variety of samples. We have liquid blood, we have swabs, we have this kind of thing. Okay, so from different material, you can, there are separate different protocols for taking different samples. Like if you have liquid blood, so the very first process of DNA extraction is to make your sample mixed with the lysis buffer. There are different steps. So once you want to extract DNA, so the very first step is to mix your sample with lysis buffer. What that buffer is going to do? That buffer will lyse cells. Now when the cells will be lysed, then your DNA will be out. So first step is lysis. Once your sample is lysed, now your DNA is there out in the liquid, but it is mixed with many impurities. There are cellular debris, there are proteins and everything. Okay, now you need to separate your DNA from that mixture, so that you can have pure and quality DNA. Okay, so now if you do it manually, it is a job of 4 to 5, 6 hours. Okay, now but what you have to do is, you have to place your sample onto machine, which is there. Okay, so you need not to do isolation, but either you do it manually or you do it with by using some machine. You have to lyse yourself first. Okay, so now the very first step is to lift the sample. Like if you have liquid blood, you will use this pipette, and whatever is the ideal requirement of machine, like if you want 100 microliter or if you want 50 microliter, so that amount will be taken out and that will be dispensed in a small tube like this. These tubes are said to be Eppendorf tube. This is having here. Okay, so these tubes are called as Eppendorf tube. Okay, so suppose, do we have liquid blood in EDT oil? Do we have liquid? आप लोगों में से कोई ऐसा है जिसको अपनी प्रोफाइल देखने में इंटरेस्ट हो? So what I would prefer कि आप लोग बजाय ये सैंपल लेने के, अगर अपने सैंपल को मशीन पे लगा के दोगे और उस सैंपल को मार्क कर दोगे, तो विल सेंड यू दैट प्रोफाइल. मैडम आपको भेज देंगी, हम मैडम को जनरेट करके भेज देंगे. विल एक्सट्रैक्ट डीएनए. जीनोटाइपिंग फैसिलिटी यहाँ नहीं है, बट दैट प्रोफाइल वी विल जनरेट इन आवर लैब. विल सेंड इट टू मैडम, मैडम विल सेंड इट टू यू. ओके, तो अब आप ये सैंपल की प्रोसेसिंग देख लो, एक कैसे लेते हैं, लेकिन जो हम लोग प्रोसेस करेंगे वो अपने सैंपल करेंगे. तो कोई एक अपने बाल निकाल ले खींच के, जिसके ज्यादा दिख रहे हो और काले दिख रहे हो, तो मेरा टारगेट ये सामने वाले बंदे हैं ना. हो जाएगा.
Okay, there is a chair for Dr. Pankaj. He can occupy the chair of.
Okay. में से किसी को अगर विश्वास हो कि ये हमारा ब्लड सही निकलवा लेगा तो उससे निकलवा ले.
हम लोग ब्लड सैंपल डेड बॉडी से बहुत बढ़िया निकालते हैं.
नहीं,
इट्स द लिविंग विच इज एन इश्यू.
मैडम ये क्या एक दूसरे पे बिल्कुल ही भरोसा नहीं है आपको? तो मतलब हम लोग गलत ही आते हैं डॉक्टर के पास.
I request Dr. Barkur sir to keep everyone in loop of what's happening and what they should look at.
All the participants are requested to be on the side of the faculties, right?
Here.
Take 200 microliters of this as per the protocol of machine. Okay, so this is how we'll open tube. Okay. This way. 200 microliters. Okay. ये ब्लड किसका था जो बड़कू सर ने निकाला था?
क्या नाम?
विकास अरोड़ा.
डॉक्टर विकास अरोड़ा.
Your registration number?
29.
Okay, so now I am putting 200 microliters of Dr. Vikas into this Eppendorf tube.
And you all will be getting FTA cards soon. So after drawing blood and putting it into the tube, they are properly labeling it, right? So the identification of the donor has to be clearly mentioned on the vial, so that there is no mixing or misplacing.
Now the next step is to mix.
नहीं सर वो वहाँ से ही नहीं यहाँ से लिया शायद पूरा पैर अच्छा ये बटन बदला हो गया
Okay. So with 200 microliter blood, we added 300 microliters of lysis buffer. Okay. It's mixed with lysis buffer now. Okay. And after every step, you need to open this and then you need to close this. क्योंकि हमको इसका क्रॉस कंटेमिनेशन भी अवॉइड करना है और इसको एनवायरमेंटल एक्सपोजर से भी बचाना है. सो ये उतनी ही देर खुली रहेगी, आप देखते रहोगे मुझे अगर ऑब्जर्व करते, मैं इसको कई बार खोलूंगा. यू वोंट फाइंड दिस ओपन कि हमने एक बार ब्लड सैंपल ले लिया तो इट इज ओपन. ओके. Now we need to add 20 microliters of protein SK, which is an enzyme.
So equal amount of enzyme has to be added. 20.
This is protein SK.
Protein. What is that protein?
What is your rating? 20 microliters.
20.
Getting into it, blood. क्या मिलाया है इसमें? ब्लड मिलाया है, प्रोटीन. So now I am adding 20 microliters of protein SK into Dr. Vikas sample. Now, after adding these two things, we will simply vertex. Vertex means mixing. So by doing this, by tapping, we will mix. Okay. Now this is mixed. The sample is ready for putting onto the machine now. Now we will have it here also. For processing here, no, the size of hair is this much. So if your root is here, so it won't be live. So that your root should be somewhere here. Okay. ये छोटा काटना पड़ेगा. अब देखिए कंटेमिनेशन बात हो रही है. अगर ये ओपन है, ये उसके लिए यूज नहीं है, डॉक्सी वाली नहीं है सर. देखिए होगा सिर्फ ये प्रोफाइल तो आएगी, इसकी आएगी, इसके ये कारण कोई और दिस शुड बी क्लीन और उसको हम एक बार वाइप कर लेंगे. अल्कोहल जो भी है ले आओ सर इसके अंदर नहीं
मैं दे दूँगी।
मार्कर पेन उधर ही
पहले बफर डालेंगे हाँ सर उससे प्रोटोकॉल लेते हैं प्रोटोकॉल कर रहे तो इसमें भी वही प्रोटोकॉल है
ओके
सेम एज
अपना करना है
ये है जो हम लोगों ने मिक्स किया हुआ है now we'll be needing buffer
lysis solution and marker pen नहीं सर
would like to put challenge on the machine let us see किसका हेयर है योगेश यूजी कर भाई जी नंबर गौतम एग्जैक्ट इसलिए होता है क्योंकि वी हैव फाउंड एंड द प्रोटोकॉल इज सेट फॉर मशीन दैट मशीन विल टेक 300 माइक्रोमीटर सो नाउ यू हैव 200 माइक्रोमीटर ऑफ ब्लैक and 300 now you have 500 millimeters. So your machine is take 500 millimeters.
20 माइक्रोलीटर के लिए कौन सी वो है? 20 माइक्रोलीटर Notice this 20 microliter was what is this 20 microliter for 1 ml?
वो डाइथायोथ्रेटॉल होता है।
ये क्या फिक्सेटर होता है कि
ये भी होता है। ये जो हार्डर सेल्स होती हैं वो सेल्स हैव डाइसल्फाइड बॉन्ड। ये बेसिकली सीमेंट के एनालिसिस में ऐड होता है। अच्छा हेयर की प्रोसेस में इसका कोई काम नहीं है। मेरे हिसाब से कोई काम नहीं। मेरे हिसाब से इसको अगर अपन नहीं भी डालेंगे तो भी प्रोफाइल आएगा। ये सिर्फ डाइसल्फाइड बॉन्ड्स को ब्रेक करता है और इसका कोई रोल नहीं है। अच्छा कोई whenever you want to put any liquid, so it needs to be like this. Yeah. This way. Okay. Now you are done with your sample processing. So we have two samples.
and your DNA profile about to come.
Okay. And what about with the? Okay. So this you need to. Yeah. Mix thoroughly by pulse vertexing and 55 or 15 minutes. Okay. So आप इसको 55 डिग्री सेंटीग्रेड पे रखना है।
It's 450 now.
Take next sample. आप लोग ध्यान रखिए ये 5 मिनट। ठीक है। हम लोग दूसरा सैंपल कर
योगेश ध्यान रखिएगा। अपने सैंपल का देखना 15 मिनट। 15 मिनट तक यहीं खड़ा रहा, हल्ला नहीं आता।
इसकी प्रोसेस ये है कि we wash it, then we dry it, and then we autoclave it. After that we take sample for DNA analysis.
इसमें क्या जल्दबाजी का काम नहीं है। You have to be cool and नहीं।
Now you have this. Now you want to cut this, and for this you use one paper. Now see, हमें सैंपल को नहीं टच करना है। हमने आप देखो कैसे होल्ड किया है। I have not touched the sample. Okay. नहीं सर छोटा सा काटेंगे, छोटा सा ही काटेंगे इसको। We take this much only, sir. In our analysis also we take this much. ये सीजर अच्छी नहीं तो इतना भी आ गया, हमारे को इतना भी नहीं चाहिए होता, क्यों उसका कहता है हाई मीडियम, इससे ज्यादा चाहिए सैंपल, ये भी ज्यादा ही है ना, वो भी काट
उससे कंटामिनेट नहीं ना, जो अभी सीजर यूज किया था उसी से काटो।
1 मिनट होल्ड करो, ऐसे ही मैं काट रहा हूँ।
तो मैं ग्लास पहने हुआ हूँ, मैं काटूँ क्या?
नहीं इससे कर देंगे ना, लाओ। यहाँ चाह हमारा लाओ ट्यूब का है जिसमें डालना। अब आप लोगों को हैंडलिंग की एक बात और बता दें, हमारी कोई भी हैंडलिंग ऐसे हाथ डाल के नहीं होती इसमें। हम लोग जब भी सैंपल निकालते हैं ना तो वी डू इट लाइक दिस, ऐसा करके निकालते हैं, सो दैट बाकी सारी चीजें हमारी कंटेमिनेट ना हो। एंड whenever you are using this, we should never contaminate this area. You are not supposed to touch this area. Okay, so we'll do it like this. We are not touching by bare hand or by anything, and with the same scissor you can put it.
Because most of the contamination are during handling.
Okay.
Now your sample is inside.
So this is swab, gauze. Again 300 microliters of buffer. तो आप डाल लो कोई भी इसमें। इसमें आपको ध्यान क्या रखना है कि अगर आप इसको बफर में ले जा रहे हो और इस टिप से डाल रहे हो, so your this tip should not touch this material because this tip is going to be inserted in your buffer. So if you touch your tip with this material, so this will your buffer will be contaminated. Okay. So if you have seen me, so how I did, I took and then I opened this and did it like this. So with some distance. Okay. Yeah, you can have it.
Lysis material. Lysis also.
ये 550 है, ये नहीं होगी। This is 20 to 200. Okay, so I have set this at 20. Okay. So what we'll do, we'll put this at 150. 150 plus 150. No, don't disturb. आपको भी इतना एक्सपीरियंस है, आप कर नहीं पाओगे, गिर जाएगा। हम कर सकते हैं कि हम वीडियो करते करते हमारी 20 साल हो गया हमें करते हैं। आई कैन होल्ड दिस, बट यू वोंट बी एबल टू होल्ड इट। इसका तरीका ये है कि इसको ये एक लिमिट तक अंदर जाती है। ओके, अगर हम इसको ज़्यादा दबा देंगे तो चली जाएगी, बट अब क्या होगा, जो 150 माइक्रोलीटर लेने वाले थे अब वो 200 माइक्रोलीटर हो जाएगा या और ज़्यादा। ओके, तो इसको एक जहाँ तक ये रूटीन कोर्स में दबती है वहाँ तक ही जाना है ना। ओके, नाउ अब एक दूसरी बात आपको बताऊँ, एक शू दुकान में सड़क में मिलता है 150 रुपीज़ का, वही शू बाटा में मिलता है 3000 रुपीज़ का। इज डिफरेंस। सो ये जो मटेरियल है इसकी क्वालिटी भी बहुत मैटर करती है कि व्हाट अमाउंट ऑफ DNA, व्हाट अमाउंट ऑफ मटेरियल वी वांट एंड हाउ मच अमाउंट दिस इज। सो दिस इज ब्रांड वाइज डिफरेंस होता है। जिससे हम काम कर रहे हैं ये कोई बहुत अच्छा नोन बहुत बड़ा ब्रांड नहीं है, ये मैं आपको बता रहा हूँ, है ना। सो हम इसको प्रेस करेंगे, नहीं इसको हम प्रेस करेंगे, ये हमने इसको लिफ्ट किया, ये आ गया। अब हमें दूसरी बार इसमें नहीं डालना है, अब हम टच कर सकते हैं इसको, ठीक है। अब विद द, हाँ डिस्कार्ड कर सकते हैं, लेकिन व्हाट आई विल डू, अभी हमारा जो मटेरियल है वो ऊपर है, और नाउ आई वांट टू डिप दिस मटेरियल इन द बफर, सो आई विल डू इट लाइक दिस, ओके। क्योंकि हमें एक्सट्रैक्ट किससे करना है, सिर्फ ट्यूब में डालने से नहीं हो गया काम हमारा, और इसके सामने बोलना अवॉइड करना है। अगर आप लोग हमें ऑब्जर्व कर रहे होंगे तो आप देख रहे होगे मैं जब बोल रहा हूँ तो इसका मुंह आपकी तरफ कर रहा हूँ, है ना, क्योंकि हमारा सलाइवा भी इसमें जा सकता है, विच विल कंटेमिनेट दिस लैंड, ओके। तो ये काम, जितना इस काम का नाम है उतनी ही ज़्यादा इसमें प्रिकॉशन भी रिक्वायर्ड है, ये भी ध्यान रखिए। नाउ आई विल ऐड प्रोटीनेस के, ये 20 माइक्रोमीटर चाहिए, और इसके लिए ये 10 वाली पाइप है, ये वाली ओके। ओके, अब ये चूंकि 20 माइक्रोलीटर्स है, इसलिए वी आर गोइंग टू डू, इसको अंदर लेते हुए लिक्विड के अंदर डिस्पेंस करेंगे, ओके। अब ये पिपेट आपकी टच हो चुकी है टिप, so we'll discard it. Now your sample is also ready. Now, for this material, there are two methods. One method is you can take scrapping of this. This is one. Okay. The other way can be you can use swabs. So हम swabs use करेंगे तो अभी हमारे पास यहाँ पर swab नहीं है.
Which one is better in each? Scraping or taking swab?
If you can take it skillfully, then scraping is better. Because अगर आप swab लोगे तो आपका हाथ उसमें सैंपल में टच हो सकता है, इसमें टच होने के चांसेस नहीं है, बट अभी हमारे पास नहीं है, तो हाउ आई विल डू, मैं आपको दिखा रहा हूँ तो आप समझ लें, मैं आपको बता रहा हूँ मैं कैसे करूँगा।
We can use blade instead of that is contaminated.
चीज़ों का इम्प्रोवाइजेशन, आपके किसी भी काम को करने के कई तरीके हो सकते हैं। हम क्या करेंगे, हमारे पास वैसे तो फोर्सअप होनी चाहिए थे, फोर्सअप नहीं है तो आई यूज दिस। ओके, नाउ दिस इज रेडी टू बी यूज्ड। अब हमको इसके लिए कोई भी एक बफर चाहिए, बफर नहीं है तो सलाइन चाहिए, सलाइन भी नहीं है तो
सलाइन
जो पानी हम लोग पी रहे थे वो ले आइए।
सलाइन ले आएगा ना मुकेश, सलाइन लाना।
ओके ओके ओके, ये तो हॉस्पिटल में चला, ये इसका स्वयं ले लेना। 5 से 10 सेकंड डाल सकते हैं, हाँ डाल सकते हैं सर। ओके, अब हम इसको इस पे से पिक कर लेंगे। दिस इज मोर देन एनफ। Okay, so now you have this sample. But I would not recommend you all to lift sample. I would never recommend. Because हमको वो काम कभी नहीं करना चाहिए जिसकी हमारे पास एक्सपर्टीज नहीं है. अगर आप उठाओगे, और अभी तो हमने कंस्ट्रेंट में काम किया, आपके साथ और भी प्रेशर रहेगा. आपने उठाया कैसे, ग्लव्स पहना, ग्लव्स कंटामिनेटेड हुआ, तो आपका सैंपल गया. तो बेटर कोर्स वुड बी, जैसे ये चिप है, तो यू डायरेक्ट इन्वेस्टिगेटिंग ऑफिसर टू कट दिस एंड सेंड दिस. नो इशू. ये लेबोरेटरी में जो साइंटिस्ट होंगे वो अपने आप ले लेंगे. गॉट इट. जो हमारा काम नहीं है उसमें हमें नहीं, हम वी आर अथराइज फॉर बॉडी, तो हम बॉडी में से लें जो लेना है. इसकी नहीं है तो आपको बता दो कि आप ऐसे ले लो या कट कर लो. इसको तो ट्रांसफर किया जा सकता है ना. जो ऐसा ऑब्जेक्ट हो जिसको ट्रांसफर नहीं किया जा सकता, तब हमें उसके साथ सैंपल लिफ्ट करने की कोशिश करनी है. अदरवाइज वी नीड ऑन टू लिफ्ट सैंपल.
ड्यूरिंग ट्रांसपोर्ट इट में आल्सो कंटामिनेट.
स्क्रैपिंग लेने के लिए आप इसी सैंपल को एक डेट दीजिए मुझे. हाँ. ओके, अब आप एक ट्यूब निकालो. स्क्रैपिंग क्यों नहीं लेते हैं उसका रीजन बता रहा हूँ मैं आपको. जब हम इसकी स्क्रैपिंग लेने की कोशिश करेंगे तो ये उड़ सकता है ना. इसीलिए हमेशा इस तरीके की सर्विसेज पे हम लोग लेबोरेटरी में स्वैप करके उठाते हैं. तो अब हम इसको स्क्रैप करेंगे तो इसके पूरे चांसेस हैं कि ये उड़ जाए. ये देखिए उड़ रहा है ना. निकाल लीजिए. ओके, ये आ गया. अब इसमें 300 माइक्रोलीटर बफर, इसको लिख लें आप लोग, जो भी इसको और इसको मार्किंग एक ही दे दें. इन दोनों का सेम डीएनए होगा. और इसमें 300 माइक्रोलीटर बफर, 20 माइक्रोलीटर ये डालिए आप लोग. ये डीएनए निकलेगा. फिर वो डीएनए आप यहाँ शायद क्वांटिफिकेशन तक का होगा व्यवस्था, और वो सैंपल हमारी लैब में हम जीनोटाइप सेंट्रीफ्यूज करा दें.
Now we will be demonstrating how to take oral to be linked to Aadhaar card. So be careful before committing anything.
तो फिर तो कंटामिनेट करना पड़ेगा इसको.
इसमें the simple precaution is that the mouth should be clean and there should not be food particles. Food particles or any stain like your bitter leaves or anything you are eating, like कैल्शियम प्रोडक्ट है या कुछ चीज खा रहे हैं ना, वो सब निकाल दीजिएगा. उसके बाद में कुल्ला कर लीजिएगा, बेहतर होता है.
As sir said, it is better to rinse your mouth before giving the oral swab.
It reduces your bacterial flora as well as the food contaminants. And after that you take a swab and on the lateral side of the buccal mucosa, rub it for 5 to 10 seconds. And then after taking it out, just dry it and then you can put the swab for the proper use.
कुल्ला करने के दो तरीके हैं, बाहर कर सकते हैं.
But at the same time, you must remember, if you are taking the oral swab in a case of oral coitus, then please do not ask the victim to rinse the. It should be taken before rinsing and preferably from behind the last molar, because that is the area which is not normally cleaned during. So be careful. Rinsing is not control sample. It's good.
Keep my mouth shut. As sir told, it has to be slightly away. It's a rotational. Yes.
So that you can not only collect the saliva but also some mucosal cells. But the another way can be you keep the piece of cotton at the opening of mandibular, submandibular gland or the parotis, so that when it
Kindly
comes
label
out,
it.
it can be
Label it,
soaked
label
into
it. Can we take one more, sir, so that those who haven't seen come? I request all those who have seen, please go back, and those who haven't seen, please come forward. So one more swab will be taken. So any volunteer? ये आपका blood तो नहीं हुआ ना? नहीं हुआ ना? आपका कुछ नहीं हुआ? I request those who are behind, please come forward and see the oral swab procedure. Yeah,
who wants to collect sample? Yes. नहीं लेने दो, हम आपके तो निकल गए, अब तो इन लोगों
Dr. Vivek will take the swab.
Why don't you collect yourself?
Vivek sir, come to the
You can
face the
collect
camera,
yourself.
please.
हाँ. And not only rubbing, you can also roll the swab along with rubbing.
रोल में कम आता है. रोल
नहीं,
में
डबल हो जाएगा ना.
empty वाला.
आपकी वाली गई, ये दूसरी है। वो उधर है। नहीं नहीं, डालो ना अंदर, डालो ना इसके अंदर।
मेरा क्वेश्चन ये था कि ड्राई तो हुआ नहीं, सर ये ड्राई तो हुआ नहीं,
अभी तो इमीडिएट लगा रहे हैं ना, तो ड्राइंग इज फॉर,
जैसे हम ऑटोप्सी टाइम का
नहीं तब उसको ड्राई करना है, बिकॉज देयर इज टाइम लैप्स इन ट्रांसपोर्टेशन.
कंपोज नहीं होना चाहिए।
या सिलिकॉन के अंदर हो ना तो भी ठीक है.
तो हम रिजल्ट सब लेते हैं, कभी कम ड्राई तो कर देते, ड्राई टप कर देते, हर ऊपर उठा रखते हैं. तो ये क्वेश्चन था, वहाँ भी मेरा क्वेश्चन
क्या चीज क्या
क्वेश्चन ये था कि जब हम ऑटोप्सी टाइम के रिजल्ट सब लेते हैं, सब लेते हैं,
उसको ड्राई कर, नहीं उसको ड्राई करना है बिफोर पुटिंग इनटू द ट्यूब. नहीं आप बाहर के कमरे में पंखे में करो ना, फैन चला के उसको ड्राई करो. क्योंकि जो द सैंपल विच इज नॉट बीइंग यूज्ड then and there, it has to be dried in air at room temperature before sealing or before packing.
Now we are going to send all our samples to this instrument. Okay. So
From there to this place.
So whoever is having
Yes.
this instrument,
What do we call this machine? Analyzer, नहीं. Extractor.
20 माइक्रोलीटर प्रोटीन के बाकी की जो सैंपल्स है उसमें सेम ऐड करके इंक्यूबेशन के लिए जाता है 55 डिग्री फॉर 15 मिनट. जो ब्लड सैंपल्स है आपके, ये अभी डायरेक्ट ये इंस्ट्रूमेंट में जाएंगे, इनको सेंट्रीफ्यूज करने की जरूरत नहीं है.
को इंक्यूबेशन की जरूरत है नहीं, इंक्यूबेशन ब्लड को. So those who are sampling blood, they need not to put into the incubator, they can directly come to the extractor. Blood need not to be incubated.
Setting up assay for 8 samples. So currently what we have set up here is 7 sample with whole blood and 1 sample with hair sample. So hair sample we already incubated for 15 minutes at 55 degrees centigrade. After that we did the centrifugation. Only supernatant was collected in a fresh tube and it is again processed for DNA extraction now. Now for each individual sample we will be setting in a particular slot, we will be setting this cartridge. This cartridge, if you can see, is already pre-filled with buffers and solutions. Okay. Also, you will find at number 1 site, you can see one adapter. If you can remove it, madam. it contains one filter. So this filter will avoid any kind of contamination or any kind of contact of our sample with the nozzle which is generating the pressure. Okay. Apart from that, we have one. Apart from that, we have one this 3 number, madam. We have one tip by which the sample will be collected. Okay. Again, place it, ma'am. No, no, no. Next. Ma'am, please take this one. No, the column. Column where we will be placing in between this adapter and the tip. Okay, ma'am, please place it. Please fix this cap in this slot, ma'am. So for each cartridge, we'll be setting up this the column, and the cap will be fitted in that particular slot so that it will not interfere with our nozzle. The same thing we have done here. Ma'am, can you please, sorry? Yes, I'll show it, ma'am, just one thing. So now, this is another, आप उसको उल्टा करोगे ना निकलेगा ये हाँ नहीं नहीं हाँ right. So you can see again this tip comes with a filter which will be used for mixing. So generally हम जो pipetting से या vortex से mixing करते हैं, it will be done with help of this pipetting. Okay, and this tip will be used only for mixing. इसको place करो. Everything is here is single time use. Everything here is provided and part of the kit. Nothing you have to take outside from your lab. Everything, including these tubes, adapter, everything. The tip, as specific, single time use, and also each and every tip or adapter is having specific site where we need to proceed because it is a robotic arm. If anyone would like to please place the cartridge, please come forward and place accordingly. नॉच है, ये नीचे जाएगा नहीं नॉच ये देखो मैडम ये नॉच है यहाँ पे ठीक है ना ये एग्जैक्टली नीचे मैंने 2 लगा के दिखाया मैडम उसको लॉक कर दो नहीं अच्छे ये ऐसे होगा सर right same with others last sorry ये second last नहीं आया सर हमको नहीं आया लास्ट वन ठीक है सर आप इसको रखो यार नाच को दबाओ दबाओ पहले उधर कट आएगी आवाज ऐसे उधर पुश करो हाँ करो ये नहीं आया ना इधर अभी जाएगा अब ये लगाना है कॉलम्स है इसमें इंडिविजुअल कॉलम ये सिर्फ ऐसे यहाँ पे जाएगा पर्टिकुलर स्लॉट में वो कैप अगेन साइड में जाएगा इसके अंदर जाएगा अरे इसके अंदर फोल्ड करके डालना है ऐसे उसको पहले हाथ से फोल्ड करो ऐसा करो हाँ अब डाल दीजिए अरे मैडम ये बता ठीक है सर नेक्स्ट टाइम आप कल एग्जाम होगा सब लोगों को अपने अपने सैंपल प्रोसेस करना है सर हमको तो दिखाई नहीं सर ये सब सबसे पीछे खड़े सर हेल्प करेंगे गुरुजी सीख रहे हैं इसको मैं डाल के दिखाऊं कैसे डालते हैं इसको कैसे डालते हैं इसको सर ये जो डाल रहे हैं अंदर इसको ऐसा बोल के कॉलम ढक्कन कॉलम बना हुआ है नंबर 1 स्लॉट है, इसमें हमने एम्प्टी ट्यूब डाला है जो कि अगेन प्रोवाइडेड विद द किट है, स्टेराइल है, जो यहाँ पे जाएगा जहाँ पे हमारा एल्यूशन जो फाइनल प्रोडक्ट जो रहेगा हमारा डीएनए, इट विल बी कलेक्टेड हियर, ठीक है, यस, इसे और ये लॉक हो गया, ठीक है, ये ट्यूब फॉर मिक्सिंग, ये हमारा सैंपल है, सॉरी सर, कौन सा वायल, वो वायल कौन सा वायल, सैंपल वाला अंदर इंस्ट्रूमेंट में रख, सैंपल का ट्यूब कभी भी बाहर नहीं खोलना है आपको, सैंपल का ट्यूब जब अंदर जाएगा तभी आपको ओपन करके रखना है उसको, एक बार आपका पूरा सेट हो जाएगा, जब आप सैंपल विल बी द लास्ट थिंग दैट विल बी टचिंग एंड क्लोजिंग द लीड, ठीक है, उसका हो गया आपका, ये देगा ना सर,
मैं पूछ रहा हूँ इनमें क्या रख, ये जो हम रख रहे हैं इसका क्या रोल है?
अच्छा ठीक है, अभी जैसे आप हमने एक्सट्रैक्शन प्रोसीजर आप जानते हैं, ठीक है, उसमें काफी सारे वॉशिंग स्टेप्स रहते हैं, उसमें फाइनली एल्यूशन बफर भी रहता है, सो यहाँ पे 3 डिफरेंट वॉशिंग सॉल्यूशंस है, एक और लाइसिस बफर है एंड लास्ट वन इज सॉरी वॉशिंग बफर है एंड एल्यूशन बफर है, सो एंड ये जो 2 एम्प्टी स्लॉट जो दिख रहे हैं दिस आर फॉर इंक्यूबेशन, जो हमारा एल्यूशन बफर है इट विल इनिशियल फर्स्ट स्टेप ऑफ द इंस्ट्रूमेंट, वो एक्चुअली आप देख भी सकते हैं, सो वो यहाँ से एल्यूशन बफर उठाएगा और ये इंक्यूबेशन के लिए लगा के देगा, ओके, सॉरी सर, एल्यूशन बफर उसका एग्जैक्ट कंपोजिशन मैं आपको बता देता हूँ सर, ठीक है, तो उसका pH 6.8 है, मैं आपको बोलता हूँ सर एग्जैक्ट, हाँ मैं आपको बोल देता हूँ सर, सो ये इंक्यूबेशन होगा एंड इन केस अगर किसी प्रोटोकॉल में अगर हीटेड इंक्यूबेशन का अगर कोई स्टेप है तो सैंपल इधर जाएगा, वो इंक्यूबेट होगा एंड देन अगेन इट विल बी ट्रांसफर्ड, तो हमारा जो सैंपल है एवरी टाइम पहले फर्स्ट थिंग व्हाट विल हैपन इज हमारा कॉलम, मैडम वो दे दो ब्लू वाला, हमारा जो टिप है वो सबसे नीचे जाएगा, मैडम ये आपको, उसके बाद आएगा हमारा कॉलम, ये ऐसे फिट होगा एंड देन एडाप्टर ये ऐसे फिट होगा, और पूरा हमारा जो प्रोटोकॉल है वो ऐसे ही इसी पोजीशन में कंटिन्यू होगा, ये जहाँ पे ऊपर में अटैच होगा, दिस विल ओनली कम इन कांटेक्ट विद दैट पर्टिकुलर बफर और सॉल्यूशन, हाँ यस 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 एवरीथिंग वो करेगा फॉर 60, वो भी इंस्ट्रूमेंट ही करेगा सर, हमको इसको कभी भी ओपन करके किसी भी सॉल्यूशन को हमारा हाथ नहीं लगना चाहिए, सो इट विल बी डन बाय द इंस्ट्रूमेंट, फर्स्ट थिंग विल बी डन बाय इंस्ट्रूमेंट इज पियर्सिंग ऑफ ऑल दिस सीलर्स, सो उसका जो कवर है आफ्टर दैट वो जो ये जो टिप है ये निकाल के ये जो टिप है वो लेगा एंड देन इट विल बी यूज्ड फॉर मिक्सिंग आवर सैंपल, ये मिक्सिंग करेगा सिर्फ एंड छोड़ देगा, फिर उसके बाद ये जो टिप है वो डिस्कार्ड होगा एंड उसके बाद ये पूरा तीनों लेगा एंड देन इट विल बी प्रोसेसिंग, सैंपल के लिए इधर रखा है सर हमने,
सर ये यहाँ पे उसका टिप रखा हुआ है जो इसमें से निकल के इससे सैंपल लेगा एंड देन इट विल कम लाइक दिस अप टू दिस, राइट, देन देयर इज अनदर टिप, वो बाकी का काम वो दूसरी टिप करेगी और ये यहाँ के डिस्कार्ड हो जाएगी, कवर अंदर जब रखेंगे तब ओपन करेंगे,
when we put inside the machine then only we will open the cover of sample and rest of the tubes containing different solutions or buffers, they are sealed with the aluminum foil like thing that will be pierced by the tip automatically by the instrument.
नो हाँ नहीं, ये यहाँ तक लिप करेगी, इसमें से करके इसमें पियर्स करेगी, फिर कुछ देर रखने के बाद इसमें आएगी, फिर इसमें आएगी, ऐसे आते आते यहाँ के वो डाल देगी, फिर देयर इज अनदर टिप हियर, तो बाकी काम ये वाली टिप करेगी, इज इट क्लियर, देयर शुड बी नो कंफ्यूजन नाउ,
सिर्फ 2 सैंपल और रिसीव किए जा सकते हैं इस स्लॉट में,
what sample is this? Okay, buckle is to be centrifuged, no? Buckle ka, no, le आओ, each sample. So we have got 1, 2, 3, 4, 5, 5 slots empty, na?
ये 29 नंबर किसका था? उन्होंने कुछ भी
ऐड नहीं किया इसमें, ना बफर किया ना प्रोटीन है, बस मत करना, बैलेंस करके चलाना सेंट्रीफ्यूज को, नहीं तो एक ही ट्यूब में चला दो वो टूट जाएगी. So one kit is meant for 10 samples, 12 samples. So otherwise it will be wastage of sample. Some room. हाँ तेरा तो लग गया अच्छा वाला, तेरा अच्छा वाला चीज लग गया. क्या है भाई वो clotting with 11 samples, right? Manish, Dr. Manish. Now the tray with all chemicals and samples is inserted into the instrument, right? And the cap of all the samples is manually opened, नहीं? We are opening ना? जो अपन ने सैंपल अभी रखे थे उसको मैनुअली ओपन कर देंगे अभी, ठीक है ना? उसको लॉक करने के बाद, ट्रे को लॉक करने के बाद so inside the instrument there are 12 robotic arms, right, which will be handling all these samples, right? So now samples are reduced to 10. The other sample was not fit for. Now tray is inserted and locked. There are locks on the side. ये locks हैं जो बगल में होते हैं ना, साइड में छोटे छोटे. हाँ, ठीक है ना? And now the caps are being opened one by one, taking care of not contaminating the other tube. To contaminate the samples from outside or from among themselves. कैप खोलते समय बहुत केयरफुल होना है क्योंकि we are opening the cap one by one. So one sample which is opened earlier may contaminate the later one, second one. And when we open the cap, there is a slot to put the cap inside so that it doesn't hang over the mouth. Yes, right? So that lid is fixed in a slot. Mask, yes, वो तो सब करना है फिर. So you have to take universal precaution while handling the samples, because not only contaminating the sample, but if there is some disease, you may have cross infection. So now we are closing the, and then there is a computerized screen. Yes. So first we have to select the illusion volume 50 microliter. Whatever sample you have. Yes. 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 So here we have selected 50 microliter and we can start it, right? It will take. Yes, you can visualize how it is acting. So now the tray is sucked inside under the robotic arms. Yes. So first step, so after inserting and closing the machine, with the start, the tray which was inserted in front is sucked inside under the robotic arm, and the covers are being pierced by the tube, by the robotic arms, right? Now it is holding the tip from the front row, then dropping it into the kit. Dropping into the kit, the last one, right? Can you see? इसको हम रख देते हैं. Now कुर्सी दो जरा. Right. So now you can see the tips are being dipped into these solutions, right? You can see the moving arms, na? Now it is taking the samples, right? First piercing the tube, now collecting the samples. हाँ उसको मिक्स करके ले रहे हैं डिपिंग. Okay. And this process is being displayed on the screen as lysis, right? Lysis display हो रहा है screen पे. Yes.
The last lecture for today, to understand the collection and preservation of samples from scene of crime, what to collect and how to collect. We have with us Dr. Nikunj Brahmbhatt sir, Assistant Director, Institute of Training at Gujarat Forensic Science University, Gandhinagar. He is heading the State DNA Division in Directorate of Forensic Science, Gujarat. He is the pioneer scientist in establishing the State DNA Lab, Gujarat, and he has also examined and helped in solving various DNA profiling cases. Currently, he is developing a kinship assessment by DNA profiling index, that is KADI software, which resolves kinship disputes and identification of suspects in assault cases. Sir, I invite you for the lecture. Thank you, sir.
Thank you. Good afternoon. सबने चाय पिया? How's the Josh? Thank you. So, since morning we are having the same subject repeating like an advertisement. So like the advertisement that is repeated over and over every time, it hammering us just to make you more and more pakka in the subject. Once again, collection and preservation of sample from scene of crime, right? Friends, as you know, processing the crime scene is very much important. The first responder and its work is very important. As we know, an automobile that run on gasoline, crime laboratories that always run on physical evidence. Without that physical evidence, one cannot help in solving the crime. So physical evidence encompasses any and all objects that can establish of scientific value, a scientific evidence from the scene of offense. We have numbers of things that is scattered around, but what is important? It is very much important. So forensic science that begins at the crime scene. Processing, if the investigator cannot recognize physical evidence or cannot properly preserve it for laboratory examination, no amount of sophisticated laboratory instrumentation or technical expertise can salvage the situation. In many jurisdictions, police agencies have delegated this task to a specialized team of technicians. But very much important, it is not necessary that in all crime scene we will definitely get any sort of the physical evidence. And at that time some other type of with evidence. Before that, if suppose is there any of the bloodstain, in a different shapes or patterns, it is very much important to look at it and to identify that what it wanted to say, and that is been known as blood stain pattern analysis. That is blood stain pattern that left by falling, projected or smeared. Blood can be analyzed by trained crime scene investigators. Careful observations and measurement of the position and shape of blood stain can give a lots of information about the direction of travel, angle of impact, and speed or velocity of the blood droplet. You see here is this picture, and that gives some ideas that what might have happened in the scene. So through this examination, we can say that the victim might be injured nearby to this wall, and that is that there is a smear we are seeing, and then there is some droplet that is scattered around, that is going to some direction. Now what does it mean? Blood stain pattern can be used to prove or refute a suspect's amount of what happened. The information from the blood stain patterns can possibly be used to reconstruct a crime, and blood stain pattern analysis can tell us the how of the crime. So how does it is helpful? The dripping of this blood, the same amount of blood drop at the different angles, and we see here the shape that is been changes when it becomes decreases in the angle, and at this point you are seeing that the point that gives the direction in which the object is or injured body is moving. Wherever we find any such sort of this blood stain pattern, we take the photograph and we write all that required information regarding it, because it is very much helpful. And then after, with this manner, we transfer the stain on a swabs. That is what we have just exercised in the adjoining chamber. So by this manner, the swabs that has been prepared, it is been allowed to air dry in a shadow. No hair dryer, no sunlight, or no any such sort of heat is permissible because that may degrade the sample. After allowing it to air dry, container is very much important. As we know, this sort of this polythene tube or polythene bag is not permissible for any sort of the biological fluid, but this sort of this paper bag, it's a good container. So always use for a biological specimen, always use a porous material as a container to keep the material enough air dried. Secondary, we have seen so many times IOs, and sometimes through the post-mortem, the garments are being kept in the same parcel, in the same bag. All the garments, if suppose kept in this manner, it means it cross-contaminate the specimen. Whatsoever the blood or any sort of the other body fluid that has been there on the garment may transfer onto the another garment, and that may sometimes gives misleading the offense. So what is the right manner? And this is what it shows. For each of the garment we have to use a separate container. For a liquid blood found at the scene of crime, we are supposed to use a sterile syringe, and we have to draw the liquid blood and then immediately transfer it into the vial which has the proper preservative. Now after transferring it to this preservative, means EDTA bulb, we need it to shake it well. Sometimes we have seen that we just transfer into this EDTA vial, but we are not shaking, and when it reaches to the laboratory, the fluid is clot, though it has been sent into the EDTA vial. So the shaking is required for proper dissolution of the preservative. Alternatively, and the best one, a clean and sterile cotton gauze or cotton swab can be used to soak up liquid blood or blood clot. Always avoid areas containing only serum. If suppose the blood that is found on the snow or in water supposed to be immediately collected through this cotton gauze and let it to be air dry. A large possible quantity of such sample should be collected in a clean, sterile and suitable container to avoid any additional kind of contamination. It is very much important. The contamination, it does not mean in the manner of bacterial contamination. The contamination may be by some other manual contamination, because while we are speaking, a micro droplet that is coming out with this air that is coming out through this mouth cavity, and that is along with saliva. So that may sometimes contaminate the specimen. It is very much important. Freeze such specimen if transfer to laboratory is delayed. That is the question one person has asked to me. This sort of different color vials that is available, the color code of the cap that is universally accepted, and as we have discussed in the morning, in the EDTA vial we are seeing here, it is a 5 ml capacity, and there is the written material is there. It has the 1.5, that is the concentration of this preservative. If suppose the wet blood stains is we found at the scene of offense or the garment, we are supposed to let it to be air dry under the shadow and not under the direct sunlight. Never package clothing or object with wet blood stains in an airtight container or plastic bag. Why? We know, because it may easily contaminate and the bacteria can grow or mold can cover that wet area of the stain. Blood stains found on immovable objects should be transferred on a sterile cotton gauze or swab and allowed to air dry before packaging. The integrity of any blood stain pattern should be preserved during packaging and transportation. For soil sample, what do I advise? Let it to be air dry first and then send it into the paper bag, but never use tin, glass, polythene type of the container. Dried blood stains. As we have seen just on the tile, the blood stain pattern should be first photographed, documented, sketched to the required extent before packaging. Dried blood stains on a movable object should be collected separately as entire item in a sterile paper bag if it is movable and easily transportable. If a dried blood stains found on non-absorbent surface or immovable objects, then they should be scraped off on a clean piece of paper. Always collect control sample from an adjacent but unstained area that act as a control sample. Fresh and dried samples can be used for DNA profiling, like liquid blood or liquid semen or fresh tissue or the dried tissue or the dried blood stain or the dried semen stain, a buccal epithelial cell, bone marrow, tooth pulp, hair with root, etc., 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 anything, right? Biological samples in crime scene need not be processed for DNA profiling if serological and microscopical methods are sufficient to give required information. For example, paternity dispute cases and semen stains without sperms. So preliminary we can screen out with the help of blood grouping, And for semen, with the help of microscopic observations of the spermatozoa, we can filter it out. In case of vaginal swabs, we are seeing this sort of this spermatozoa under microscope. For each of the animal, the shape and characteristic structure of the spermatozoa is very specific, and through this microscopic observation, we can easily identify the species of animal. This is a presumptive acyl phosphatase test. Friends, we are the student of science, and for performing any of the test, we started with presumptive test. So at the scene of offense or even in the laboratory when we opened the case, we started with the presumptive test. This is acyl phosphatase test, and here we see that is a seminal acyl phosphate and vaginal acyl phosphate. Both are showing presence of acyl phosphatase, but this is more concentrated, this is faint and lighter one. The concentration of acyl phosphatase is more in semen compared to vagina, but that is possibly helpful for presumptive test only to confirm that it is possibly a seminal stain. It is not a confirmatory test, right? Use a glove for collection of biological specimens. It is advisable to collect as much biological sample as possible for DNA profiling from forensic exhibits. It's on your discretion that what does it mean as much as possible, right? It depends on the nature and size of the sample you are getting, and on that you have to send to us. Collection of biological samples for DNA profiling should be left to an expert in order to maintain purity of the sample. To maintain the quality of DNA from blood samples, it is recommended that EDTA is added as an anticoagulant, and if police personnel is not able to collect the sample in a short time, please preserve it in a freezer compartment of refrigerator, or if suppose the deep freezer is available, store it at -20 or -80 degrees Celsius. Extraction of DNA from fresh sample is however recommended for better DNA yield. Tissue samples, both fresh and dry, should be kept in cool and dry place or may be kept in a frozen condition. If dry blood stain or semen stain cuttings from garments are to be sent after a few days for DNA profiling, they may be kept in a desiccator and then packed in paper envelopes and transported. It is important one. Samples like dry bones, teeth, hair, etc. may be kept in a paper envelope and sent. But if suppose you are having a less quantity or less number of the hair fibers, and if you send it through this Ziploc bag, polythene bag, then it is also permissible. Why? Because in the morning one speaker has shown that slide, and there is a question why it is there in this polythene Ziploc bag. Because it is less in number, and in suppose that in the laboratory there is a ceiling fan that is going on, and the person opening that paper fold, that may blown out. Just not to lose that important evidence, if you kept conditioner plate supposed to be totally dry, air dry, and send it in a Ziploc bag, polythene bag, then it is okay. Liquid blood from the scene of crime may be collected on a clean piece of cloth, dried and sent in a paper envelope. All blood stain exhibit like clothes should be dried and packed in such a way that the stains do not touch each other and the stained portion does not contaminate the unstained portion. This can be achieved by covering the stained portion with clean paper. Separate items of clothing must be packed separately and properly labeled. So this is one demo here in picturesque manner. This is a stained garment having a stab wound over there, the stab cut mark is there, And you see here this garment that has the step mark, the cut mark. The clean paper is overlap, then it is fold, and then after it is placed in an envelope. This is called a manila envelope or printed labeled envelope of the different sizes. I also advise my police officer, police personnel when they come for refresher courses, and I even give you the same advice to you. If suppose you may keep this sort of this different size of the pre-printed material paper envelope, it is very much helpful for you people for packing and forwarding the garment, and even the biological specimens too. There are polythene bags having the button, what it says, the button packing material is there, so that gives the safeguard to this glass container or the glass test tube from breaking. So that sort of this envelope is also available in market. Blood, soil, grass, hair, etc. should be dried thoroughly in air and kept in paper packets. Unstained soil should be collected as a control. Stained knife, axe, bullet, lathis, etc. should be secured separately in a wooden or cardboard boxes with the stain areas covered by paper after drying with cotton wool. You see here one weapon or a knife having the stain smear and how it is been packed.
Interrupt for a while.
Yes, please.
We are saying that everything, every sample, every article clothing, we would like to segregate separately, and paper would be the best form for packing. Whether it be knife or clothing.
Whether it is?
Knife or clothing or.
Yeah, for knife I am showing you how it is been wrapped and to sand. Cotton wrapped, and then with the help of this paper, it is wrapped, and you are saying between the two thermocol sheet, it has been pressed, and then in a cardboard box or a hardboard box, it has been sent. So that is the right methodology. For each of the type of the specimen, like the article like bullet or a bit. Say for a bullet, it is made up of a soft material, copper and tin. If we do not handle it with care, so many times you might have observed that from a body you need to remove that bullet. So that bullet you need to take out with due care, that with the forceps even it will not produce an extra scratch mark over it. So with the delicacy you have to taken it out, and under the cotton wool you have to wrap, and in the matchbox you have to sand it. So that is the ideal protocol theoretically. लेकिन जैसे सुबह सतपति साहब ने बोला कि व्यवहारू भी होना है, तो उस समय कॉटन वॉल में रैप करके आप पेपर एनवेलप में भी बेच सकते हैं. सो दैट डिपेंड्स कौन सा आर्टिकल है, कैसे हम पाते हैं और इसको किस तरह से भेजना है, वो हमारे खुद की सोच पे जाता है, समझ पे जाता है. So that is the methodology how to send these articles. Post-mortem blood should be stored in ice cold and should be sent immediately to the laboratory. If delay is unavoidable, blood may be taken on a piece of cloth, dried and sent in a paper envelope. Cement stained clothes should be dried and packed in the same manner as blood stained clothes. They should be sealed properly and sent. So this is the sealing methodology for parcels and for envelope, and that was the end of my session. So I do hope since the very beginning of this seminar from morning to this, I do hope that is embossing well in your mind? That? Yes, please.
Excuse me, sir. Sir, is there any database where we can look upon for packing these articles? Because once we come upon some articles only, we can identify.
Yeah, I have already sent my draft to Madam Jayanti ma'am, and she will definitely share you. Is that? Health center, community health center, and health center of these districts in our state. So no, not yet.
Preserving tissue or organ or fetus, on the last presentation we have seen that we have to use sterile glass containers. Can we use sterile plastic containers?
Of course, yes. For a tissue, if suppose you have, I am not asking for more quantity. If suppose you are having a 50 gram, that is sufficient for our laboratory. Send it into this append-op tube, that is what you have just seen in the adjoining chamber, right? Place it into the ice bucket, send it immediately to the laboratory without any preservation. I welcome it.
Sir, in most of the cases you have told that it should be air dried under the shadow. I have seen one device in forensic medicine people are selling called forensic evidence drying cabinet. Is it effective?
Of course, yes, it is effective. It is vacuum chamber dryer, so it is helpful.
Sir, I would like to ask one thing. As you mentioned that each article should be packed separately in a separate container. So suppose we have received a body and that is wearing 4 or 5 clothes. So we need to pack each cloth in separate
Yes, sir. We have to prepare a separate package, separate packages for 5 garments, and all that 5 packages in one container, then it is okay.
So having the 4 or 5 clothes into one
that is what I shown. That is what I shown. Yeah.
Any other question from the audience? I have some questions. इसलिए मैं पूछूंगा मैं last में पूछूं. Should I ask before you? I have some practical this thing. My question is to Dr. Bhatt, Dr. Pankaj and everybody.
Yes,
what
sir.
happens when the doctors prepare vaginal smear? They usually prepare 1 or 2 slides, which are initially sent to the biological division before sending to DNA. So if they are stained, how far they are useful for DNA extraction if they are stained? Because they are not only stained but also covered with cover slip.
Sir, you are preparing smear with the swab. So
Slides.
you are preparing smear with the swab, and you are definitely send that swab to the laboratory for further analysis. And 3 slides you are preparing. 1 is for your laboratory in hospital, 2 are for forensic laboratory, right? Of the 2, 1 is for the staining, and another one we take it as a preservative. If suppose from the swab we are not getting the good result, that smear may help at that time.
Because in our state also, we have a clear-cut guideline that we receive 2 slides in our state. Okay, so one if the case is for biological analysis, biology and physiology, so only 1 slide is stained, and there is a clear-cut guideline from DNA laboratory that always keep 1 slide unstained for further analysis if required. So whenever examination, DNA examination is needed, that slide is used.
But let me tell you one to extend my answer in the. I had just got one case from Assam CBI, and that was the stain slide. And the protocols are now like the computer software. There is a computer that is we are seeing that it's always been updating. In DNA we are also always getting some updation in the protocols, and through the stain slide I am successfully extract out the DNA. Though I get the partial profile, that was the limitation, but I got it.
Second question, that you said if there is a small government object, it can be preserved as a whole, right? If it is small. Say, there is a big room, and there has been a case of gang rape. Somebody did in this corner, that corner. The floor is porous. The stains are not visible. Then what do you suggest? What they should do, or the
person who is examining.
Sir, actually this is not the work or the means headache of this forensic medicine department, but for general knowledge and just to give the how the forensic scientists trace out or detect this sort of the stain from this porous material, like the वो क्या बोलते हैं वो गाय का इससे मिट्टी वाला जो फ्लोर होता है, हाँ, yeah. So in a dark we are using ultraviolet radiations. So with the help of ultraviolet radiation it fluoresces out. The seminal stain that fluoresces out, and that fluorescence it is helpful for us to have the probability of that seminal stains, and we at that time do some presumptive test. If it gives some presumptive test positive, we collect that material and send it to the laboratory for further examination.
Right. So first it has to be visualized, no? Then,
Right, sir.
So either by lamp
Multi-
or by spraying some fluorescent dye.
Multi-tunable lights is available, sir, and multi-tunable lights
This is
gives
what,
a
they
different
should know,
way.
na?
Yeah,
I know
exactly.
it, this is for them to know. Any other question, please?
My last question.
Okay.
योगेश भी पूछना चाह रहा है।
अगर किसी क्वेश्चन डॉक्यूमेंट में कोई बायोलॉजिकल सैंपल है तो उसको कैसे एक्सट्रैक्ट करेंगे, क्योंकि क्वेश्चन डॉक्यूमेंट के लिए भी स्पेशल गाइडलाइन होती है कि आप उसको कम से कम डिस्टर्ब करें, तो अगर उस पर है तो उसके लिए क्या गाइडलाइन है?
गुड क्वेश्चन। क्वेश्चन डॉक्यूमेंट में अगर मान लीजिएगा कि इंक से लिखा हुआ कुछ बनावट है, इसके ऊपर कुछ लिखाई है जो इंक से है, अगर मैं इसका स्वैप बनाना चाहता हूँ, हो सकता है वो इंक स्प्रेड हो जाए, बराबर। सो एट दैट टाइम व्हाट वुड आई डू, और मेरी लेबोरेटरी क्या करेगी? हाँ, सबसे पहले वो जो डॉक्यूमेंट है, वी आर हैविंग द वेरी सोफिस्टिकेटेड इंस्ट्रूमेंट्स इन माय क्वेश्चन डॉक्यूमेंट डिपार्टमेंट, एंड दैट इज कॉल्ड एज VSC 5000. So with the help of that instrument, I put that document into that, and with the infrared rays, I will try to decipher whatever there it is been there, that is been writing and what I wanted to have. Then after I make this photography, okay. So all that the writing material that has been preserved and documented. Then after I prepare a swab from it. How to prepare a swab? We have just learned.
How much this part we can?
How much? That is just from the beginning of this session in the morning we are learning that just a pinhead 1.2 millimeter size of the stain is enough.
This was purely from forensic science to forensic science.
Yes.
He came to document from the body. He moved from the body towards the document, which is very interesting to us. My question was, how much is the workload on an average on the state forensic science lab or the Bhopal forensic science lab? What I mean is, like, is there like we quantify our work or experience or things on the type of a number of cases that we kind of do? Do you have some kind of I don't know, and any benchmarks, or we handle so many specimens, like yeah, you have 2,000 cases a year, or we have 250 of poisoning, or we have so many sexual assault, or
for receipt of case there is no guideline, any number of
Yeah,
cases can be received,
yeah.
but for the output there is
Yes.
a clear-cut benchmark that all the scientists in our state, for example, if you talk about MD, everybody has to do 20 cases per month. All the scientists are supposed to do 20 cases minimum.
20 cases of DNA per month, per scientist.
Minimum. Maximum they can do as much.
This is minimum.
And no cases means n number of samples with one case.
It can be n number of samples. You may have, the Ramkishan was telling me, he was in one case he was having 70 exhibits. Even then he has to do 20 cases. Yeah, that will be counted as a single case. Because your target is to do 20 cases. In the last you will be assessed on the basis of, once your CR is getting ready, it will be based on your total output. So nobody talks about exhibits.
Is that just your lab, or would
It's more or less entirely, there is almost same scenario.
It is actually debatable thing. It is not been nationally guideline. Yeah, nationally accepted, or there is no any such guideline. Yeah.
For
state
MP,
labs.
this is, the thing is like this. For
It all depends on the controlling authority.
Controlling authority.
What he is expecting from this.
Oh, there's another question I have. When we send a sample, will it go necessarily to the Bhopal Forensic Science Lab, or we can send it directly to state, or?
No, that depends, madam, that depends on the investigating officer and the facilities available at different labs.
That is already decided. For these many districts, this lab will take care, and for these many districts, this lab will take care. This is
So it
already
will go to
decided.
the Bhopal lab if Bhopal lab has
Yeah,
that facility.
like from Bhopal, Vidisha, Ujjain, and maybe 4-5 more districts. These will deposit cases for analysis in RFSL Bhopal, and for other some labs there are they will deposit at Sagar. Then we do have a RFSL at Indore. So there are certain districts assigned for that lab. Then we have a lab in Gwalior also. And like now at present position, we have only DNA facility at Sagar. So all the cases from let it be from any district, it will go to Sagar only. But maybe in the lab is finalized, and we'll start DNA facility in Bhopal also within maybe 1 or 2 months. So then we'll be having again a division that these many districts will send DNA case to Sagar, and these many districts will send to Bhopal. So this will be all the DNA case will go to Sagar lab.
मेरे मतलब गुजरात की बात बताओ, हमारे यहाँ ब्लड अल्कोहल में जो भेजे जाने वाले जो सैंपल है, जैसे आप जानते हैं गुजरात इज़ ए ड्राई स्टेट. ये हंसने की बात नहीं है, कहने की बात है, कहने की बात है. Gujarat is a dry state, and we are having a numbers of samples for analysis, and for that फॉरेंसिक मेडिकल या जो मेडिकल ऑफिसर जो है जो ड्रॉ करता है ब्लड सैंपल, इनको डायरेक्टली ये सैंपल लैब में भेजने होते हैं. बाकी जो सैंपल है, जो आपको विसरा सैंपल लेने हैं या जो भी केस के रिलेटेड सैंपल लेने हैं, वो आपको पुलिस पर्सनल के जो मैसेंजर है इनके जरिए लैब में भिजवाने हैं. सो हाँ, फॉर योर नॉलेज इट इज़ बेटर टू हैव द जोन वाइज demarcations of the laboratories. As we are having in Gujarat the 2 centers for the DNA lab, Gandhinagar and Surat, and so the Gujarat is divided in 2 zones. Say in the MP you are having right now the 1 center. So accordingly, but it is being done by this police personnel, right? So you have to just hand it over your specimens with the required documents to the police personnel for laboratory analysis. But I do hope through the discussions of the today, you might be pakka in how to collect and preserve the specimen, right?
One more question to Dr. Pankaj to highlight something about touch DNA.
Touch DNA is something fancy. Okay. तो हमको कुछ करना है, और एक बात होती है थ्योरेटिकल और एक बात होती है प्रैक्टिकल. हमने कहा कि हम ये इलेक्ट्रिक स्विच है, इस पे एक बार हाथ लगाएंगे और इससे प्रोफाइल आ जाएगी. हमने कहा कि हम ये पानी को मुंह से लगा के पी रहे हैं, इसमें से आई कैन जनरेट माय प्रोफाइल. ओके, बट अगर हम ये कहें कि कितने लोग इसको कर सकते हैं, तो वो गिनती के होंगे. तो दिस डिपेंड्स ऑन स्किल, क्योंकि टू डील विद अ गुड क्वांटिटी ऑफ डीएनए सैंपल्स, इट कैन बी अ जॉब ऑफ एनीबॉडी. But if you have to deal with a minimum quantity, then you need a very skilled person to handle. So if I say, yeah, it can be done. No doubt it can be done. I am holding this mic, my profile can be generated. But if I hold it for some time, not like कि इसको हमने ऐसा पकड़ा और छोड़ दिया, कुछ आने का नहीं. होने को तो theoretically ये भी है कि there are certain publication कि एक बेडशीट पे कोई सोया हुआ है एक नाइट में, from that bedsheet a profile of that individual can be generated. There is publication. Okay, but if we say कि हम किसी को भी देंगे और वो उससे प्रोफाइल जनरेट करेगा, तो आई विल से नो. हर कोई नहीं करता. स्किल अपनी जगह है, वो आप लोग भी सहमत होंगे, आपकी फील्ड में भी आप लोग जानते होंगे. तो हर आदमी कर पाए जरूर नहीं है, बट यस इट कैन बी डन.
गुजरात की बात बताओ.
This is the mortuary of the New York Medical Examiner, where if you enter, the condition is you will have your sample, the DNA done on your sample, which will be kept in their record. Only then you enter. Otherwise, you look at the mortuary from the glass panel. And the logic is something similar to that. In case you come inside and if you happen to touch, or I don't know, breathe, or your DNA, if it is around and we are collecting some sample and yours is contaminating, so in the database we will have yours, so we can remove that this person was just visiting, is not related to this dead body or the suspect on the...
For the knowledge of all, all the laboratories, like in my lab or in Sir's lab, whosoever is working, their profile is already there. We have typed all the samples of all the laboratory persons, and those samples are kept in our...
It is mandatory.
Are the visitors?
No, not for visitors.
Yeah, this is for visitors that they have.
They don't allow visitors in the working area.
So they ask for that for every visitor who wants to just walk into the mortuary.
No, there are certain things you can cover this. You can.
लड़की को बाहर खुले में खेत में ले गया। दो लड़के थे, एक लड़की थी, इनके साथ रेप हुआ। इसके बाद इसको अगवा करके सूरत होते हुए बॉम्बे ले गया। बॉम्बे से वो लड़की को कोई न कोई मतलब रेस्क्यू मिल गया और वो वापस आई 1 सप्ताह के बाद। आफ्टर 1 वीक सी अप्रोच टू द पुलिस पर्सनल, और उसने इनके साथ जो हुआ जामनगर से वो पूरी बात इसने कंप्लेंट के दर्ज करवाई। 7 दिन के बाद हमारे वहाँ डिस्ट्रिक्ट का जो फॉरेंसिक ऑफिसर है, इनको साथ लेते हुए वो जगह पर जहाँ विक्टिम ने बताई वो जगह पर इनको ले गए। वहाँ से हमारे फॉरेंसिक ऑफिसर ने लड़की ने जो बताया था कि ऐसा हुआ, बाद में ऐसा हुआ, बाद में ऐसा हुआ, वो काम करते करते इनको जो प्यास लगी थी तो पानी के पाउचेस भी ले आए थे लोग। पानी के पाउच से इन्होंने पानी पिया था, 2 पाउचेस। वो 2 पाउचेस, और ये लड़की का नसीब अच्छा बोलो या जो भी बोलो, वो पाउच वहाँ पे थे उस समय। वो पाउचेस विद ड्यू केयर फॉरेंसिक ऑफिसर ने कलेक्ट करवाए और हमारे लैब में भेजा। सर, पाउच से अगर पानी कोई बंदा पीता है, किस तरह से पिएगा? एक कॉर्नर है, वो दांत से काटेगा, वो मुंह में डालेगा, सामने वाला जो पोर्शन है उससे वो प्रेस करता जाएगा, पानी अंदर जाता जाएगा। ठीक है। द पोर्शन फ्रॉम वेयर द लीप इज कम इन कॉन्टैक्ट, आई गॉट द प्रोफाइल ऑफ विक्टिम, एंड द पोर्शन फ्रॉम वेयर द पर्सन हैज हैविंग द प्रेशर ऑफ द हैंड, दैट फिस्ट, वहाँ से एक्यूज के प्रोफाइल मिली है। दैट इज टच डीएनए सर, राइट। दूसरा एक केस बताऊँ मैं। एक आईपीएस ऑफिसर, इनकी लड़की घर में अकेली थी। इनका फादर या नॉर्दर्न ईस्ट एरिया में ड्यूटी पे था। इनकी मदर जो थी कोई सोशल रीजन की वजह से बाहर गई हुई थी। लड़की ने अपने फ्रेंड्स को घर में बुलाया था। 12th स्टैंडर्ड में पढ़ती थी वो। रात को 12-1 बजे सब इनके फ्रेंड्स चले गए। लड़की अकेली थी लेकिन केयरलेस थी। ये भूल गई कि मैंने मेन दरवाज़ा जो डोर है मैंने बंद नहीं किया है। वो जब सो गई, जैसे लैपटॉप पे देखते देखते या मोबाइल में देखते देखते, सब कुछ चालू था, लाइट वाइट सब कुछ। जब सो गई तो उसको लगा कि इसके छाती पे कोई बैठ गया और इसको कुछ मतलब ट्रैंगुलेट करने की कोशिश कर रहा है, नाक दबाने की कोशिश। वो तुरंत जागी, धक्का लगाया। उसने देखा एक लड़का था जिसने अपने चेहरे पर वो माइक्रो फिल्म जैसा एक पेपर बैग आता है, बराबर, इससे एक मास्क कवर बनाया हुआ था जिसमें 3 होल थे, 2 आई के, 1 नोज के, बराबर। लड़की ने इसके पीछे तुरंत भागी और मुंह देखने के लिए वो खींचने की कोशिश की। वो इसके हाथ में आ गया कवर, पॉलिथीन बैग। लड़का भाग गया। पता नहीं लगा कि वो फ्लैट से उतर के कहाँ कैसे गया। वो जो जगह है वो आईपीएस ऑफिसर, आईएएस ऑफिसर के रहने की, ऐसे हाई रैंक गवर्नमेंट ऑफिसर्स की क्वार्टर्स थे। और वहाँ पे बाद में फॉरेंसिक ऑफिसर, डॉग स्क्वाड, सभी को बुलाया गया। डॉग स्क्वाड ने काम तो किया, 3 मंजिला नीचे उतरा फिर वापस आया, 3 मंजिला नीचे उतरा फिर वापस आया, कोई क्लू नहीं मिलता है। 3 मंजिले नीचे एक दूसरा आईपीएस ऑफिसर रहता है, वो भी हाई रैंक का है। लेकिन वहाँ से किसी ने अपने जो सर्वेंट्स हैं या इनके जो मेंबर्स है इसकी सैंपल्स देने के लिए कोऑपरेट नहीं किया। हमारे पास वो सैंपल्स भेजे गए। अच्छा, अब ये वाक्य जो हुआ था, ये हड़बड़ी जो हुई, इसमें वो लड़की के थंब में और थोड़ा सा ये स्क्रैच हुआ था ये थ्रोट के पास। तो ये थंब में जहाँ पे स्क्रैच हुआ था वहाँ पे दीवार पे स्मीयर था पैटर्न, वो स्वैब लिया। वो पिलो जिससे इसने स्ट्रेंगुलेट करने की कोशिश की वो कवर हमने लिया, बराबर। और बाद में वो मास्क जो लड़की ने खींच निकाला था, कवर पर से जो प्रोफाइल निकली वो किसी लेडी की थी, दैट इज़ नॉट मैच विद दिस विक्टिम लेडी और विद एनी मेल पर्सन, ठीक है। वॉल के ऊपर जो स्मीयर पाया गया वो लड़की का खुद का पाया गया, और मास्क जो बंदे ने पहना हुआ था तो इसकी इनर सरफेस जो है मास्क की वो स्किन के साथ कांटेक्ट में आई है, ठीक है। वहाँ से हमने स्वेबिंग करके जो प्रोफाइल निकाली तो वो ऐसे आदमी की निकलती है जो पुलिस ने जितने भी 30-40 सस्पेक्ट किए थे अरेस्ट, किसी के भी साथ वो मेल नहीं खा रही थी। नाउ द केस इज बिकम वेरी सेंसिटिव। Attack was on the IPS officer's daughter and then it comes to my neck कि पुलिस ने 40 को किया है और तू ऐसी कैसी प्रोफाइल लेके आया है, किसी के साथ मेल नहीं खा रही है। इट मीन्स कि कोई नहीं कोई तरीके से ये जो केस की प्रोफाइल निकलती है डॉटर की कैरेक्टर के ऊपर कुछ एलिगेशन कर सकता है कि डॉटर कुछ छुपा रही है या कुछ फैब्रिक स्टोरी बना रही है। तो साहब बात तो वैसे देखा जाए तो सीएम तक पहुंची थी और बोला गया था कि अगर अब की बार अगर कुछ नहीं मिलता है देन यू विल बी टर्मिनेट आउट, किक आउट। लेटर मेरा नसीब अच्छा है भाई साहब कि जो तीसरी मंजिल नीचे जो आईपीएस कोऑपरेट नहीं कर रहे थे उनके यहाँ जो सर्वेंट रखा था उनके घर में वो सर्वेंट ने चोरी करने की कोशिश की, और वो कोशिश में वो आईपीएस की वाइफ ने उसने देख लिया, उसने पुलिस को बुलाया, तब जाके उसने कोऑपरेट करने की कोशिश की और बोला कि हाँ हम देने को तैयार है। तब तक तो वो मना ही कर रहे थे। वो जो इनका सर्वेंट बंदा था इसकी डीएनए प्रोफाइल के साथ वो मास्क की डीएनए प्रोफाइल मैच खा गए साहब, और तभी जाके एक केस सॉल्व हुआ कि वो बंदा बारी बारी से, क्योंकि ये लड़की केयरलेस थी, दरवाज़ा खुला छोड़ती थी, वहाँ के चोरी करके चला जाता था। लेकिन वो रात इनको अकेली पाकर इसका दिमाग कुछ बिगड़ा हुआ था। तो दैट इज कॉल्ड टच डीएनए मैडम। तो
it cannot be done by anybody. This is damn sure, isn't it? It's not a job of anybody. ये बच गया। अब ये देखो निकालने दो प्रॉब्लम है, ये निकाला ये इनकी स्किल है, वो इनकी तारीफ की बात है, लेकिन निकाला तो मुश्किल में भी यही पड़े। टच डीएनए दोनों तरफ से नुकसान है।
So yeah, of course, yes, it is relevant. It match 100% to that tag.
इसलिए मैंने इतना अच्छा सवाल पूछा समझो।
Definitely.
Yeah, it would.
Because touch DNA is mostly in common places, not in specific. If it is a house, you touch the hand.
Raped and she got pregnant, and her mother went to the local doctor for a termination of pregnancy, criminal abortion, for the criminal abortion, and that criminal abortion done there. After that, she got infection, internal infection, and case was slowly became serious. Then referred to the MOI hospital in Indore. And there, due to the severe infection, uterus removed by the gynecologist. And after 2 or 3 days, she died. And her body sent for the mortuary for the postmortem examination. And that case, after 10 to 15 days, how we stabilize the paternity? By the we have only the body of that female. Fetus is not available. Fetus is not there. And that rape was done by some local gunda, like that some person. That family was poor family. So that there was problem for establishing the paternity. There is any clue for establishing the paternity in that case?
There is no evidence. How could?
There is no starting material.
No evidence. How could we get? No, it is not possible. Thank you.
So I want to ask,
What happens with the sample post-analysis? Means what is the protocol for handling or discarding the samples after you have done analysis?
There is a guideline.
Because we are seeing a number of samples.
The post analysis, whatever the remains is there, that is, we send it back in a sealed condition to the police personnel, and that is being deposited in the respective court.
Just like weapon. Just like weapon.
Just like weapon. Even the empty glass vial.
It was very useful and very interesting and very informative lecture, and I'm sure even when we are late by almost an hour, I don't see any tired faces over here. So thank you so much, sir, for that enlightening lecture. And with that we come to end of this today's session, and because we have so much to learn and so little time left, I request you all to be here again tomorrow on time, because today it was a bit late on our side also, and which has been rectified today. So from tomorrow we will be starting sharp 9 and breakfast from 8 onwards. So I request you to be on time so that we can use maximum of it. Thank you so much.
The admissibility of DNA in court as evidence in India by Dr. G.V. Rao, sir. About sir, he was the first chief scientist of laboratory of DNA testing appointed by Government of India at CDFD. He holds a doctoral and postdoctoral degree in DNA testing. His contribution in various cases like the Amur case, Jharkhand Mukti Morcha case, wildlife poaching cases makes him distinguished amongst his field. After serving the government by providing DNA testing services, he has now taken up a new career as an advocate in areas of criminal and medico-legal cases trials across the country. I would like to invite sir to deliver his lecture. Sir.
Coincidences. My first publication was with forensic medicine professionals. Yeah. So when it came to doing dissertation for part fulfillment of MSc, my guide then suggested, why don't we do some occupational health hazards by microbial agents which can affect forensic medicine doctors while working on autopsies. That was my first international publication. So many coincidences. Now I'm starting the talk, but first I like to ask my delegates to take out their notebooks, and please note, first thing is, go back to your mortuaries, buy a 3-liter pressure cooker and keep it ready. That's the first point you should make, because we are talking of sterility, sample collection and all, but when you do not have autoclaves in minimum setup, the best would be a pressure cooker which could be used to sterilize your material before sending it to the labs. Excuse me, can you show me? Next slide. March made all the difference with the first publication of Alec Jeffreys, who was working on myoglobin gene, and he discovered that some parts of the human DNA were varying from person to person, but was following Mendelian form of inheritance. And this publication was first tested on a Ghana couple who were seeking custody of their child for immigration purpose. And this is the way to start it. This was called as multi-locus fingerprinting, or RFLP method, the first generation DNA fingerprinting that is seen on the right side of my slide. Immediately later, our own Dr. Lalji Singh, who was already working on hypervariable regions in humans on the Y chromosome, he published in 1986 in Human Genetics, means it's about a year and a half time after Alec Jeffreys, that he showed that BKM sequences, that is called banded crate minor satellite sequences, are polymorphic in humans and can be used for DNA testing. What Alec Jeffreys did was he patented his technology with Life Technologies then, and it was available to us at a cost which was very expensive. Lalji being Lalji, he developed and got it patented with CSIR, and this started our path on DNA. That's why he's called father of Indian DNA fingerprinting. And they give quite a good power of discrimination, and also they can be used for ancestry and ethnicity studies. I don't want to go into this. I know yesterday we were really on collection for several. So this is how blood is collected. This is very field method. Take a vial, drop it. If you don't get anything, you can use heparin to rinse the syringe. Don't use heparin as a thing, but you can rinse the syringe on field work and use it to collect blood, put it in ice. Yeah, normal saline is what I tried during my PhD days, was found to be the best available, fieldly available preservative. You can buy that in any medical store, but fill it up only 1/3 of any glass container. And formaldehyde and alcohol is a strict no for DNA testing. This is done yesterday. Earlier days we used to get all the material, the whole bed sheet, mattress and all to the lab. But you can simply scan that or put it under UV light and pick up the relevant portions and send, because now sensitivity is increased to picogram level. This is how you pack it. And right. As per the request of Dr. Jayanti Yadav, she has asked me to elaborate more on the DNA bill, being now on the other side of the bar, that is as an advocate. This bill was in line since 2005. And earlier to this, there was one NABL guidelines which were published by Government of India, which unfortunately nobody followed. And I think Abhay spent 1 year on that, right? Our colleague, along with me and Moinak, he spent 1 year compiling the data and providing the guidelines and all. But unfortunately, it was a recommended document and not a mandatory document. So many labs don't want to follow it, because then the quality control becomes very high. So then that remained like that, and then 2005, Lalji, Dr. Bhargav, all luminaries of molecular biology, they started work on this DNA profiling bill, as it was called earlier. That went on till so many years, and thanks to veterans like J.M. Vyas sir of Gujarat lab and others, this has ultimately become an act in February 2019. What does this bill say? I'll speak more on this because this is what everybody of us are interested to know. It regulates the use of DNA technology for identity of persons, and then it also provides for establishing a national data bank and regional data bank to have this something. मेरा चेहरा नहीं है तो ठीक है. So it creates a crime scene index, suspects index, offenders index, missing person and unknown deceased. Okay, this so far so good. Then it also provides at national and state level, and who is going to deposit those samples? They are going to be deposited by state forensic labs and central forensic labs. And then these labs will receive the DNA, analyze it as per standards, and then share the data to DNA bank. Now here you have to pay attention. The DNA profile, the DNA sample is still with the forensic lab. Only the data is getting transferred, and that is going to a national repository. The bill doesn't provide where it is going to be established and who has access to it. They have said yes, one button search key will provide, all good. But whether the police will have access, whether the private person will have access, the bill is still not clear on that. This data can be used for identification of persons, well and good, even civil matters like paternity cases, inheritance matters. These profiles also will be uploaded. But their consent is Article 21, which is right to liberty. I have my own data, my own individual, I am a self, I am a person. I don't want to share anything of mine. So this was prior to this bill becoming an act. In July 2017, this case is going on between called Justice Puttaswamy versus State Government of India. where he said is Aadhaar card information should not be shared. Aadhaar card is only for a kind of personal security or personal identification. But the promoters of this bill were in such a big hurry that without giving consideration to the larger bench of Supreme Court, they went ahead and released this bill and it got passed. Whether accreditation of DNA labs will be taken care of the repository or the board, not yet mentioned yet. So who is going to say which lab is following standard procedures? FSL of one state will use a Promega kit, another person might use a high media kit, whether these loci match or not. So there is no single scientific procedures laid down as per this bill. So I might use Promega, Pankaj might use China ka kaun sa? Pan Global. Then it goes haywire because the repository is tuned, the server is tuned to receive data in a certain format. And if I don't submit as per it, it might get rejected. So that has also been not taken. I'm not being skeptical or I'm not being, but what I'm saying is this particular bill was made without participation of forensic professionals. People who work on the bench, people who have published on national databases, who have published population data, they have all not been considered. Who constitute the board? Please read here. Experts in the field of biological sciences. So who will be the biological sciences? One person from neuroscience, one person from Malbio, somebody from virology. It should have been experts in the field of DNA sciences or forensic sciences. That provision. And Director General of National Investigating Agency. He could have been given a password to access the data. He need not be on the board. Then directors of CBI, who will always be an IPS officer. I hope no IPS is here, no? Okay. Then CDFD director. This was there when Lalji Singh was there because he was a hardcore DNA person. But after him came Dr. Hasnain, he is not a DNA person. Then came Gauri Shankar, he is a virologist. Now, what's his name? Devashish Das. He is not a DNA person. Now what will he contribute? They have never deposed in a court of law. You put Article 21(14) sub means barabar. So these people and member of National Human Rights Commission, these people are going to say who is going to deposit, how it is to be accessed, how the bill is going to work. So this is one thing. Then they also said these are the people who are going to give accreditation to DNA laboratories. So every DGP would like to have his FSL accredited, whatever so manner. And then developing modules for training manpower. This is the pointer, ma'am? Excuse me, this is the pointer?
Yes.
Front one.
Ye wala? Now, who will develop modules for training manpower? Not people like Brahmbhatt or Pankaj Srivastav or Mainak Banerjee, no. It will be these people who will be developing modules for training manpower to deal with DNA related matters. Now, what is the way it is going to work? Any laboratory which conducts DNA testing will establish the identity of individuals as listed in the schedule to require to obtain accreditation from the board. They will give that board may revoke accreditation for every 2 years. Here the case load is 3 to 4 years. So the time he receives is accredited. By the time he releases the report, he is removed from accreditation. What will happen to the case? It fails. People like us who know DNA and law together is sufficient ground for us to crush the report there and then itself. Then they say sharing of DNA data bank is what the bill provides for. Now in criminal cases, the laboratory is required to return the biological material, which mostly they don't come to take it. In my experience, they never used to come back. They used to take the report, leave the exhibits with us. We never used to send. So the investigating officer will deposit the DNA profile with DNA data bank, and other laboratory, they will destroy the sample and inform the concerned person when it is not collected. Suppose the case reaches to Supreme Court and Supreme Court orders reinvestigation, and then you won't have DNA sample. This simple aspect of a lay person has not been thought of by the enactors of the bill. Then who will removal of DNA profiles is what the bill provides for. There is no mention who will remove it from the laboratory. The laboratory is the generator of the profile, and they will still keep the DNA profile with them, but it will be removed from the national server. So that has also been there, and the best part they always keep telling is one-time keyboard search is what is required. You don't have to do a lot of computation, and that will take care of the bill. Now these are the things which briefly I'll go through. It will cover DNA technology, it will include civil matters, consent of individual is not specified for DNA profiling in civil matters, storage of DNA profiles is provided for, it may include information other than identification, removal of DNA profile is very vague, removal of profiles related to missing persons is only on written request, no requirement for DNA laboratory to remove the profiles. This gives them all over powerful control over the profiles. Written consent may be inadequate because of the pending judgment of the court. Collection of DNA sample from, they said you can collect DNA samples from photograph or video. This shows the ignorance of the board. I'm being candid about it because how can you collect DNA sample from a photograph or video unless it's touch DNA? That too in a crime investigation where it is part of evidence. Then comparison of DNA fingerprints and DNA profiling has been made even today when latent fingerprint technology is always on the wane. So why this particular DNA bill was not sent to a standing committee and why it should be now sent to a standing committee is what we have to discuss. Now what is relevance to Puttaswamy judgment? This particular Puttaswamy is a retired justice of Supreme Court, very old man, about 90 plus. He appeared party in person in Supreme Court, challenged the validity and implementation of Aadhaar card, and it was referred to a larger bench. What he said was Right to Privacy Act, and this particular person went up to Law Commission and made them stop making further recommendations on privacy and sharing of data, and then in the conclusion part of this particular 2017-18 bill, they say providing provisions intended to protect the right of privacy but subject to outcome of the judgment in Puttaswamy case. Till such time consent is not available to the bill to make the database. The Sri Krishna report is still to be released. It says that providing a framework for data protection, privacy, and laying down foundational principles on how public-private entities must be treating an individual personal data. The Law Commission Bill of 2017 says it can be used for 13 codis system. When did 13 codis disappear? 96. No, 2016.
2001, 1st January 2017.
17 it disappeared. Now we are working on somewhere around 25 to 27 loci, and these experts so-called have noted that it can be used on 13 codis system as a way of protecting privacy. I just fail to understand, and I don't know how the audience feels about it. What has codis system got to do with protecting privacy? Or are they trying to say that 13 can be retained or kept secure, the released 12 or 14 can be released in public domain? That we don't know. The bill is not clear on that. But all the people are very happy that the DNA bill has been come into enactment, which is not proper. All said and done, is our law enforcement agency ready to take up the provisions of the bill? A big sorry, no. Because they don't have everything has to come from the Home Ministry or from the other enforcement agencies. Their bill has not noted how the local police will interact, collect samples and give it as per the provisions of the bill. The consent part, the collection, laboratory, codis, he won't understand. Thanadar will not understand all that. So there are risks of contamination, risk of chain, they are all not been subjected to. Worst part is, this DNA bill has not provided for technological guidelines. That 1998 guidelines has been forgotten. This bill says we are going to constitute a working committee to look into the technology as per the persons involved in the board. So the board persons who have been given opportunity are not from DNA background. And they are going to suggest technology, they are going to tell Pankaj, Brahmbhatt and everybody how you have to do DNA testing now at this stage of life. So that part has not been discussed. And they have a provision for including advocates, lawyers and all. And definitely there are all the Supreme Court, the lawyers will go into it. The working committee will take another 5 years. So what is my concern is, immediately they should form, keeping DNA experts together, form guidelines of how to do DNA fingerprint, how to follow, what procedure. There will be uniformity of testing. It's not that I come to Sagar, it will be one way, I go to Jammu, it is different, I go to Gujarat, it's different. No. It has to be standardized. And then Home Ministry has circulated a set of guidelines recently, which says investigators officers should search crime scene and scientifically collect, store and transport DNA sample in crime cases according to the bill. But the bill doesn't tell anything about collection, storage and transport of DNA. So there is a lacuna there. So had it been referred to a standing committee of experts, these things would have been addressed. The last part is how much it is going to cost. The allocated budget of the state government is 20 crores only, which is not sufficient to run one DNA lab in one state. And they say 20 crores, and the bill under RTI which I obtained, the bill money spent for making this bill itself runs into 5-6 crores. Only the committee meetings held in very exotic locations, they have spent 6 crores to make this enactment, but overall for the whole project they are giving 20 crores only. Whereas 5 years back, United Kingdom spent 3,000 crores then of Indian money to bring a similar kind of enactment. So with this kind of situation, I don't think the DNA bill is going to provide anything for us in the future. With this, I close this DNA bill part and move over to some recent developments, how it helps medico-legal professionals in precise post-mortem interval with entomological evidence. Now we are into only pupil growth or the life cycle of the particular fly. Now there is an accuracy has increased to estimate post-mortem interval on the developmental genes in forensically useful purposes, species. This is more of science, so I think people like Moinek Banerjee can take a lead on this to provide some technology for this country. Flow cytometry is performed, and cell suspensions is run on a Coulter cytometer. Histograms are generated which show the progressive denaturation, which will help you to fix accurately how time since death can be calculated. The other method is progressive deletion of 4,977 base pair deletion of mitochondrial DNA, which will be useful for estimating age at the time of death. This can also be used by people like, even from forensic medicine department can take up with collaboration with other Malvaya labs. It's a very good project work maybe for students for their dissertation. Now I come to the legal part of it. Any questions up to now? That means it has gone over? No. No, I mean from the delegates.
They said the deletion of data can be done with the consent of a missing person.
Yes.
So who will give the consent?
Suspected relatives. Suspected relatives.
No, I think it's when he is known, then that data can be removed. Something.
No, this part itself is very vague. You know, who will delete?
Gray. All gray areas.
Yeah, you may be also right, and you know, the bill also might be right. But I prefer you be right. I prefer you be right, because Yeah, once he's found, there's no need to put him on that. But then the data will still remain in the laboratory.
I think very interesting point is that anybody can interpret its own way.
Yeah, yeah,
That's the quality of the bill.
that is democracy.
Democratic, more democratic.
So this is, UN says that paternity test is the best way to prove parentage. But what our law says, now from here on we have to pay attention, is so many contradictions at the apex court level itself. In this particular, I think interested people can note the citations and read it later. It is 2005 in Supreme Court 285, 27th April publication. High Court should have held the lower court that the conclusive DNA test would have provided necessary material for an effective adjudication, is one judgment of 2005. Now, what are the legal strategies to obtain samples for DNA testing? He may be asked to voluntarily submit a DNA sample, which was done in ND Tiwari case. He refused. And then a court determines there is reasonable doubt, he can provide that he should authorize law enforcement to collect a DNA sample. Then section 53 of CRPC authorizes a medical examiner of an individual such as arrested on charge of committing an offence, believing that such examination will afford evidence or abandonment. The suspect gives up control and possession of an item that contains DNA, for example, cigarette butt smoked by a suspect, discarded, or a semen stained undergarment, he gives it on his own, and that the suspects abandoned item could leave the immediate area. A detective can recover the items and send for DNA testing, is what section 53 provides for. Then coming to this particular judgment, he says, the apex court again says, in every case one has to put DNA evidence in context of the rest of evidence and decide whether it should be taken as a whole. It does amount to prima facie case. But the point is, to create a DNA profile you need a suspect. Then comes right to liberty, right to privacy, and then 53 section. So how can a person's blood sample or a tissue sample or whatever, salivary sample is collected and then create a prima facie evidence against himself? That is incrimination. So that part has not been addressed. This is Article 23. This helps all the earlier cases where it goes against the earlier cases. He says, no person accused of it shall be compelled to be a witness against himself again. In the past, they say every case one has to put DNA evidence in context of the rest of the evidence and decide whether taken as a whole, it does amount to a prima facie case. In the Article 23 says, no person can be compelled to become a witness. There is contradiction. That's why the conviction rate is low. It's not because of backlog of cases as you were saying yesterday. It's because of law.
I'm not talking about backlog. It is because of use of non-appropriate technology for, like they're using.
It has been in mind for some time.
Hi, Mike.
Is there in law something like where does your personal liberty end for public justice? Is there
Article 21.
Is there a distinct that particular article does that apply to when we say DNA database? That it is my right to privacy, it is my right that this my thing goes. But whenever I get caught in a situation,
consent.
Yeah, so then my private thought will take a back seat and the public or
the law takes over.
Yeah,
that is what is right to liberty. Right to privacy is total restriction on yourself. Nothing can touch your liberty. That was not addressed by the bill, that where his consent ends and where the authority takes over. The bill did not address that. They said subject to the outcome of Puttaswamy judgment of the larger bench, which will decide the boundaries of end of the privacy and beginning of the public domain.
And is there a Privacy Act in the making which is close to completion?
That's still pending with Law Commission. Okay. Privacy Act.
Okay.
It's Law Commission is looking into it.
Which supposedly would define a lot of boundaries
That was a recommendation of the single bench in Supreme Court in the Aadhaar card case. They referred it to the Law Commission to review its decision. They said it can't go beyond constitutional. You need constitutional amendment to do it. So that Privacy Act is stuck there. No,
that has to be read with other articles where consent has to be taken. And when you are taking the consent, it should be there should be clear mention that what are the sample you are likely to collect.
No, they say bodily sample.
Yes.
No, that has been amended thanks to Ram Jethmalani.
But without consent, you can't collect certain samples.
No, section 53, 54 has been amended. I am coming to that slide very shortly.
Okay.
Because Dr. Jethmalani, who appeared as a defense witness in one of the cases where appeared in Swami Premananda case, which he himself underwent training in DNA testing for 6 months and came to cross-examine. At that period of time, we developed a very good bonding. No doubt the case ended in conviction and later, conviction and then, and such other tests which is which the registered medical practitioner thinks necessary in a particular case. Then section, no, I won't go there. Still the guideline for deciding paternity, section 112, which says that genetic fingerprint conducted with the consent of that man in case of the child by permission of the court is not the father. Court is satisfied under the test that the test under subclause can be conducted. Law part, I will come to other judgments later.
2012 amendment.
These are all raised. 2005 also, sir. 2005, 12, latest. So this was the first case where Lalji appeared as a witness, the groundbreaking of DNA testing in this country, where paternity was established, and also section 51 of that period of time it was discussed, and it ended at granting maintenance for this case. Such time, Lalji started DNA somewhere in 87. Till 1995 this case came out, government was just not inclined to think about DNA testing or promotion of it. Only when their own Congress leader was implicated, he was sent to jail, and then that period of time they released the first grant of about 6 crores for establishing DNA lab, the first type of lab in the world at that period of time. That's how this is called the foundation stone of CDFD. This part could have been avoided, but they said that unless the DNA tests are proved to be scientifically accurate. This was a case where Jethmalani did, where this case also went. It went into very deep because there was a defense witness expert from abroad, Dr. Wall, who had appeared. But unfortunately and good for us is that person forgot to sign the report, his own report. So that was held to be a major lacuna in the case, and his findings and his chief examination was set aside.
And I think it was a cyclostyle.
So, it was a manual typewriter.
In this case, there were 2 DNA reports, one defense expert's DNA report.
Yeah. Yeah, Premananda was supposedly a migrant from Sri Lanka. He was not a Swami, but you know, in India, whoever wears saffron clothes, except Yogi, others are all considered saints. So this person started an ashram in Pudukottai, of a small place in Tamil Nadu, and he used to say that I am going to look after, he himself was a migrant, but he said I am going to take care of abandoned Sri Lankan population people or their children, and then he started to misuse that position. He was sexually harassing and he was also into that. And whenever a girl became pregnant, they would be taken to an own hospital within the campus and aborted. And then what happened is one girl escaped from there and she went to a police station and she was protected immediately, and her fetus was collected. That fetus was sent to us, but Premananda refused to give blood sample. But later on, ultimately at that period of time, the High Court pushed him to give the blood sample. We tested, we proved that he was the responsible for the product of conception. DNA test was done, report was submitted. Jethmalani entered, he said, I don't rely on this, I want the sample relied on by the prosecution for retesting. That is the first case where Madras High Court held that part of the sample can be given to the defense for retesting. He collected that, and you remember, Mainak, you were there, 98? Yeah. So they came with big force, with their foreign expert and stuff like that. We gave them whatever under seal. We had nothing to hide. They took it to US, they got it tested. Then they definitely, as expected, against us, they said, no, it is not clear. Then he put the report here and he took leave of the court and went to London, underwent training, came back. Then he started questioning BKM probe, the one which Lalji developed. Things like population. See, till 2005, when screening population was done, screening of population was done in Arizona, two people with same, unrelated people with same 13 loci were found in 64,000 population. Then the emphasis on population data increased a lot. Till 2005, not lot emphasis, because we had a very limited population data in those days, in 98. He said it is inadequate, he brought in authorities, and I remember it was a huge court hall, old palace converted into it. And this gentleman had a very good, he already has a very good, the personality was very good, thundering voice. And his senior most junior would be about 60 years old. And they would come with crates of books to, as kind of intimidate us. And he would come rack up everything and he used to put all the bookmarks. And then first time Lalji went, he said, what is your age? And Lalji was quite innocent man, frankly speaking. He was not street smart, he was a very transparent person. And he just declared his age, 47 or something. He said, that's less than my experience. So Lalji kept quiet. And then this person started removing one book after the other, one book after. And Lalji said, there's several hundreds of books on molecular biology, DNA testing. How can I say this is correct, this is wrong? And he got furious. Then he started kicking around, he kicked the chair, and then, this went on for 8 times. 8 summons were issued for Lalji. Lalji went 4 or 5 times, he got sick. He said, I can't go from here to Chennai, then take a train to Pudukkottai. Okay, police was cooperating, but this guy played all kind of antics in the court. And this wall was sitting in the court. Every question he would go, ask him, again come back. Delaying process. Every session used to end 2-3 days. And then this person, then he said, no, this is all wrong. I'll now produce my report. Then Lalji said, no, see, there's Dr. G.V. Rao who worked with me. I'm going to send him. You take, court allowed. He went to High Court and said, no, G.V. Rao's name is not mentioned. Then, because Lalji stated an oath, so it was taken, I was allowed. And we went and he saw me and I was young, definitely, at that time. So he said, your boss himself is less than my, what should I tell about you? I said, fine sir, carry on. Then he said everything, he produced his report, I report, my report, everything went on. Then at the end of the day, I told the prosecutor, this report doesn't bear signature, cannot be relied upon. So immediately he jumped to his joy. He went to the court and he said, ma'am, this report cannot be relied, it is unsigned. That time he remembered that he didn't look at the report properly. It was just 3 pages of manual typewriter, maybe typed in Chennai or Delhi. And no name of the expert mentioned, no signature, just a bland statement that the product of conception DNA profile does not match with that of the accused A1. That's all is a report of the foreign expert. And he got so wild and he literally abused, used very worse language against Bhanumati Justice, who is now present in Supreme Court. Then she asked him, then he came out. Then while going, he gave his visiting card and he said, Dr. Rao, next time you come to Delhi, meet me in Rashtrapati Nilayam. He was trying for presidentship that time. To why recently, of late, DNA reports are being set aside by trial courts. First of all, it's accreditation problem. Qualification, expertise, skill and experience is insufficient for the expert to depose evidence. This is one thing which state governments are not thinking, neither are any regulatory body thinking is. A person working in biology cannot automatically become DNA expert because there is a slot available for his promotion in the DNA section. He cannot. He doesn't have the skill and expertise. When we put him, now I know, when we put him in the box, he just doesn't understand the terminology, the spelling. So that is one of the reasons DNA reports are being set aside. Procedures are not as per standard practice. This is what should have been mentioned in the bill. That is not taken care of. And nothing mentioned about probability. Even now you ask, they say 1 in 30 billion, 1 quadrillion is what latest witness told me in Agra. He said 1 in quadrillion. I don't know how he came to that conclusion. So then validity of procedures is not properly mentioned. They are in question. No data at all. That's what I've been suggesting to my younger colleagues is, please pull all your data, make it yourself. Don't wait for any regulation. Pull it yourself, make it available so that any lab does, compares it and comes to some probability match, which will increase the evidential value of the DNA. No doubt I am now on the other side of the bar, but my heart is still there. So I always prefer the DNA report should be as strong as possible, that nobody should be able to challenge it. But they are very lacunae. And then the procedures are not properly followed. Court rejected DNA evidence is flawed and PW17 is not reliable. That laboratory should expand. This is not, this case, this particular last paragraph was not mentioned either by the prosecution or the defense. It was observation of the court itself. He said it is expected that the laboratory, unfortunately it came, this report is referred to CDFD report after I left, would expand its analytical capacity to include mini STR testing and mitochondrial DNA for fingerprinting of decomposed samples, which will enhance the accuracy and acceptability of its report. So the judges are learning more and more, but unfortunately the scientific fraternity, excepting you people, they, no, otherwise Pankaj will say, Guruji, aap aisa keh diya, waisa keh diya. So they are not enhancing their knowledge. You can't blame them because they are loaded with casework. They don't have time to write papers also. In fact, I think you took leave to write paper, right? Bangalore case, this is also interesting case. This is also against Swamiji. Unfortunately, I put many Swamijis behind bars, which my BJP fraternity may not like it, but yeah, BJP hai kya? Abhi nahi hai. Chalo. Swamiji, who calls himself as Srimad Jagadguru Shankaracharya, Sri Sri Raghaveshwara. But I don't know, he got involved with one Srimati. And this Srimati had relationships and she kept the cement stain clothes better than a forensic lab. Well preserved. Really ma'am, my experience is she preserved it for 4 years and then started blackmailing this person. Then it was subjected to DNA testing. He was not arrested. He was, I mean, charge sheet, FIR was registered, not arrested because of his connections with the ruling party. That person who reported is a wonderful case. It could have landed him in a conviction. This is a person of forensic science laboratory from toxicology background who's been promoted as a DNA expert. His Dr. Purushottam, I think he did some PhD from some Kadapa University in Andhra, which even I was not aware. Underlined portion, this portion. In the cross-examination, yes, this is the judgment part I have extracted. He has clearly admitted that his interpretation number 3 of DNA profile of saliva of cigarette butts are incorrect. Therefore, set aside. Further, he goes to say, I myself can't read it. Have not been reported as it would have matched with the DNA profile of the accused. Because it would have matched, he has not reported. That means he is helping the accused to go away. And then he says, yes, DNA of some locus are degraded. Therefore, testimony of that PW3 23, one cannot deny interpretation is vague and not conclusive. He has also admitted in that his interpretation of the DNA profile.
Left to left.
Yeah. He says that his interpretation of DNA profile of saliva and DNA profile of chunuban, or I don't know what is incorrect. His interpretation is incorrect, that the DNA profile of saliva and DNA profile are identical, and from he himself says in his report and he himself contradicts.
Why the prosecution is fine?
No, DNA. Yeah.
The statement, the one that, you're mentioning that he's saying that this is incorrect and I didn't do because it could match. I was just wondering about this language of when we have this court procedure put in text
when they ask us a question and we reply as forensic expert, which I think would be with all scientists, everybody who's asked question, the question is put like, would you think that this report is not correct?
We say false.
And we say yes, yes or no. You know the way how questioning is put, and then you know how they say, or the typist or the judge gets written in terms of it. I mean, here in the text, it always looks so as if that person has said, yes, I am doing, you know, I didn't do it, I did it wrong, I didn't do for this reason. But it's the way.
That is material admission, madam.
Yes.
In legal terms, that is material admission and
Yes.
contradiction of a statement. Now we put a question.
Yes.
Say your interpretation of profile number 3 is wrong, is inaccurate. What do you say? False. False. You can add, witness adds. You add, it comes into the record. But here his statements were very clear. I did not include the profile, some locus loci of the thing because it would have matched with that. It is not a question answer. It is not as simple as false, true or correct. He elaborated on it. He said, I didn't put it because it would have matched with that of the accused person.
Yeah.
That is material admission.
Yeah, yeah. What I think I wanted some of the students, PG students who go to courts with us to, you know, to take a note of the fact how things get written, how they are asked, how we are supposed to deposit. It is such a mix of science, technology, art and observation. But my God, how it looks finally is such a different story of when you are doing it, when you are right there as the first person.
Yeah, we write it in first person. This is the
Yeah.
judgment part with the judge,
Yes.
right?
Yes.
But these are all material admissions. You can't deny it now. He had said it is inaccurate, false. It is accurate. That's it. That meaning will come in the judgment. Here is very, very specific. That's why it is a very sensational murder case where 12 accused got out. It was brutal murder case where a family was eliminated by 12 persons from some state who came here to conduct that Supari. They say no Supari. They took Supari and killed the family. And here cigarette butts were found, bloodstains were found because of the lack. What I'm trying to say with emphasis is lack of expertise and lack of deposition skills. This kills the case.
This is the lack of integrity.
Yes.
Yeah, this is lack of integrity.
Dr. Rao, my question is whether this false statement was the admission in the court or was and contrary to his written report.
No, his admission in the court. It is his deposition.
But was it contrary to the report what he submitted? DNA report?
Because the questions were asked why certain loci are missing, he could have said they didn't amplify. Partial profile. Something he would have said. He said I did not include them because it would have matched with that of accused person.
So he admitted.
He admitted. But he was honest to admit it. That we should appreciate.
GB, in fact, what I think he was also trying to ask is that is quite often, and everything has been written down, right?
Yes.
And when the right and at the end of the cross-examination, you have to sign that.
Yes, you have to read and sign.
And you have to read and sign that. But what happens is, this is almost 10-page, 20-page document
Maybe,
and all that.
maybe.
And we were discussing just before the talk, is that in Kerala, the judge actually writes by hand there. And if his handwriting you are not able to understand, and if you start interpreting something else, and you are also in a hurry to actually get up from this particular mess, you hurriedly sign it and go off there. And hopefully that the judge must have written down what we have said it. But that is where it becomes extremely important that you should actually should be given some amount of time to sit at leisure and read the entire document thoroughly, and then
Before you leave the court, before you leave the court, you are not in a hurry to leave. You are in a hurry to leave. That's why you are leaving.
And this is what actually happens. Particularly for all the medical fraternities who are actually in a hurry to leave the court, and they say, let us get rid of this mess there. And that is where actually such
like Lalji was. Like Lalji was always in a hurry to come out of a court first, because he was a person of a particular stature which none of us, or maybe in future, can match.
Actually, this would be how you are describing the court drama he was put through. Looks like a beautiful movie material to learn for people. And I don't know whether anybody knows of it or not.
Not
no
many.
reason why anybody should get curious or go ahead and know. It is absolutely wonderful the way things he is being put through and how he stood through them. It's really, I think that life is
I mean,
learning.
often we all agree we are insulted or humiliated in court. Right. That's what I avoid during cross-examination is I respect him because I was in his position few years back. But we are humiliated. They put several kinds, sometimes they become too personal also. But you see,
that's the difference which I thought it does not happen at all to, I don't know whether it's just me or us, but I find that in the court, I get the maximum respect that I get in society. You know,
till you enter the box and witness advocate comes in front of you.
No, even then, I have noticed it is actually, it is the most gratifying part of my career that, you know, there are 20 people listening and they take down and they ask properly. Even the cross-examination when it is there, I thought it was wonderful, but you know, and harassment I thought was something which a doctor, if he was incompetent in terms of clinical practice, you know, in age estimation cases or something that we saw in files, that I noticed, yes, this person who came before me was harassed, but he did not know how to address the age report well, and which is why he probably got harassed. But experts, like you mentioned, have been harassed, and you feel.
Phone up in the middle of the night and say, what Venkat, you said something, this is something, I think this is the right way, what you're saying must be a very bad code. Because here it's so disciplined, I know a person looks into microscope and says DNA in India. Like Monica was saying. And poor people like Pankaj and Brahmbarji, they keep testing all kinds of ways to find whether DNA is present or not. You know, this kind of things, but I think you are lucky enough not to be put to that kind of situation, because it depends how a defense advocate looks at the expert who's coming, whether his or her evidence is crucial to the case or not. Okay. Right. If suppose your report is not so crucial, even not, sometimes non-cross examination is the best strategy. Yes. Chief examination finds a no cross. That is the best way. But sometimes when the crucial witness, like we all went, the only evidence was DNA. So we are harassed, humiliated, so that we break down at one point of time and we start admitting to all his fallacical theories.
Yes, whenever the witness is crucial, the intimating factors of
Increases.
the defense does, yes, it increases to a larger amount and it tries to put you down that way.
Yeah,
because
that we
scientifically
all-
they can't put you down.
Right.
So
Scientifically
they can't put you down. So these are the tactics they take over.
In fact, what happens when you or me, any experts vote, they feel that advocate is very learned, he is very senior, he is well experienced, so we start shivering in the court. Rather, we should have a feeling that we are the experts, not they are not.
We shouldn't appear to be scared of
Expert.
them.
We should not appear scared of them, but we should appear that we are expert, they are laypersons, number one. When the questions are asked, you should be very attentive, and what answer is being given by you, you should be very watchful. You must see the screen on which it is typed, or the language is spoken by the judge. If you have any objection, you can say, no, it should be written like this.
Yes.
So we are
No,
just
that
in
part
hurry
I
to run
think
away from the
Dr.
court,
Banerjee-
and when we don't bother what is being typed on the.
No, I think Dr. Banerjee did make a very good point that we should always read what we
Yes,
have.
that is the last, but we
because
should be
recently I
watchful
went
even
to
during
a
typing.
court in Bankura where the deposition was in English, and the horrible English that they had written, I had to almost spend 45 minutes reading my statement, the 4 pages statement I have deposited, and it took me almost 45 minutes to correct the spelling mistakes and all lot things, and especially so when they ask the double negative questions. So that you have to be very careful when they are ऐसा नहीं हुआ था, it was not there, so you have to be very careful whether you are actually what you are going to say is being typed rightly or not.
I think one very important question which I got, sir, this training. I think this is very, very important mentioning all these things because all our doctors needs to be trained to go to court.
Yes.
In a proper way. No. To take their own self-confidence.
Right. To observe as observation.
As an observer, and they should understand that why this practice, pressure tactics and all from lawyers and others, because there is the money involved for them. They want to win the case by any means. And as you very rightly pointed out, one of the law minister who got this thing all admitted, got the funds, he used all the tactics against Dr. Lalji Singh also, because invited Dr. Lalji Singh 2013 here, and he talked all these things.
Yes.
He said it was a very bad time for me.
He refused to go.
Yeah.
Then I also told him, because I said, sir, they might come with some available warrant, non-available.
And Ram
He said,
Jeth Malani-
"Or, Venkatesh, I am not going. I
Very
can't
good.
take that kind of
We had a
insult."
very good reputation about Ram Jethmalani, but it
No,
all
after
maligned.
that- no,
It was
after that
very
he
bad.
lost. After that he lost everything. So these things I'll pass through because my colleague is waiting for his presentation. Can I borrow some time of yours?
Mine most of
So here it was mistake of CDFD's scientists who succeeded me, where they came up that, the prosecution has failed to come up with any explanation that no tampering has occurred with most material exhibits of the case. SPR Prasad on record proved tampering with the material exhibit. That's how they got acquitted.
Tampering is after collection or before collection?
Before collection. No, after collection coming to the lab. Tampering they admitted happened because they said, this case, as you said, I should discuss some salient point of this Nupur Talwar case. There was a pillow cover, crucial evidence, which was this guy called Krishna. This exhibit was found in Krishna's house and was sent. So definitely it would have linked him more with the crime scene, how the pillow came there and all. Reports were submitted, time was there. Then CBI, when the trial is about to start on DNA part, CBI gets the report to see. Their legal team advised them that this can, get them acquittal. So they write a letter, very specific letter. We also used to get those letters which would find its way in the dustbin. But CDFD took up very great interest in this letter. They said, are you sure whether no mislabeling has occurred in your laboratory on the exhibits returned or examined? Please verify and tell. This letter comes in August. Within the same day, the person who brings the letter is an SP of local CBI. Immediate reply is given. No time taken to verify records, photographs, nothing. Immediately director issues a letter. Yes, you are right. There has been mislabeling of Krishna's pillow cover, which has been rightly corrected now. Exhibit X so and so may be read as exhibit this so and so. The whole case turned upside down. Actually, these people should have been prosecuted for perjury and misleading the court, but I don't know, the parents also left that case after that, they got released. And then the second point was there was no possibility of any error as claimed by prosecution having crept in the description of the two material exhibits of this case. That is pillow cover going from here to Talwar's house and Talwar's house to Krishna. Therefore, and also they proved there was a typographical error in the description of pillow and pillow cover from the room of the victim. Now what more can an expert commit mistakes? Therefore, such a sensational case, even today we don't know who the murderer is. There are two cases which are ghost cases. One is who killed Arushi, other is who drove the car in Salman Khan case. These two cases puzzle all the forensic people today. They found it relevant for further examination with a scale and put it on a white background, and that is missing nowadays. I don't know what's the reason now. There are no longer photographs accompanying from the mortuary to the police to the lab. That would also help in strengthening the case to show what was the measurements at the time of postmortem, what was received is one and the same. And here also they can repeat the same thing. That will fortify. Now because of lethargy or caseload or lack of promotions in job, I don't know what frustrations.
Few centers do maintain
Those standards? I've just lost track now. I don't know how long I've not seen such kind of photograph.
Could be that nowadays we are not prepared previously for getting some sort of photograph. Previously what was there that we have to call a photographer, or we used to call our own this camera. Right now we are not prepared because we already have a camera in a mobile, right? So whenever we see some good thing, we immediately click. right? So it's not a preparedness is lacking.
No, but that should photograph with a scale, and you know, you sending it helps us as a scientist, or rather help them.
Like the next case.
The query actually.
The Pushkar case is all about the cameras and the photographs.
Yeah, cameras and photos, all electronic evidence.
I just one query. Madam has just raised one point. Taking the history from a case, from any victim or an accused. So recently I got one question from IU as well as in the court that you are not authorized as a doctor to take the history. Just this is one statement was given during my this cross, and as well as the same I got this same statement from one of the IU. He said that, sir, when you take a statement or a history, it becomes a statement.
Yeah, it becomes statement.
It becomes a statement. So I just wanted this sort of clarification. So are we not authorized to take the history?
No, you are provided with FIR copy.
No, not about FIR copy. Man, a victim or
No, you are talking about medical legal cases.
Medical legal.
You are not supposed to take any history from.
Sir, this is one of
History from the
medical examination.
No, police you can take. From other sources you cannot take history of the case.
Police, from the case, from the victim or an accused. I am talking about when they come to us.
It is inadmissible. It is inadmissible.
No, it might be inadmissible.
But once you record a statement as a government doctor, it becomes part of the record, which can go in contradiction to the earlier statement recorded by other authorities. Therefore, only police or under 164 magistrate.
So then, when we got into the training during our internship or all our MBBS curriculum, we were taught that this is one of the part of medical examination. Whenever you examine a patient, you start off from the history.
Yeah, that is our medical part.
No, sir. But that's
We are guided by medical. You see, the medical examination done by medical officer is guided by the medical principles, not by the legal one. And you see the history. It depends whether court gives importance to the history or not. It depends on the judge. But medical officer, unless he takes proper history, his examination cannot be proper.
1 minute, sir. What is the requisition given along with the person sent to you for medical legal cases is only what you have to comply with. There is a strict red boundary line there. You are requested to collect the blood sample or examine the victim and provide samples if collected so to the bearer or IO. You have to restrict yourself only to the part of requisition only. You cannot cross that boundary and take history. नहीं आप बैठ के बात कर no problem sir.
You see, we have got national protocol issued by Government of India, Public Health Department. There is clear mention that doctor should record proper history. Unless we record the history, our examination can never be proper.
That is government of India says. What law says? Law says what was the requisition given to you?
Law doesn't say that history should not be recorded. There is no
recorded by IO only.
No, there is no mention how a medical officer should examine the victim.
Because you follow Government of India guidelines, we follow Constitution.
No, but there is no mention that you should not take the history. History should not
Constitution is above. So you are infringing the rights of the victim. You are asked to do examination of the victim.
Sir, I will give you one reason.
Please.
One reason, sir. Until unless we take the history, so there is one biggest lacuna is many times we fail to identify few injuries. If I talk about the
No, you are talking of 376 cases,
no,
right?
not only 370. Any case, any medical legal case, may it be injury, may it be drunkenness, may it be this accident or whatever cases, there is injury. Until unless we ask the history, until unless we take some sort of statement from the victim or an accused that आपको कहाँ दर्द हो रहा है, कहाँ चोट लगी हुई है, so he or she has to explain in her own language, right? So जो भी वो बोलता है हम उसको उसी भाषा में लिख देते हैं, right? So
No, let us break this question in two parts. Now you stop here and tell me. No, that will be second part. So first part you are saying kahan chot lagi hai, she shows. What do you write in the statement? The victim showed.
Not sure.
That's it.
As per the yes, that's what we write. As per the statement given by such and such person, she fell down from the such and such area and she had experience of these injuries assessed, or she had been.
But law doesn't allow you to do it.
Many times we miss the injuries. There are multiple injuries over the body. Some may be paining severely, so the less paining areas might be left. So that has to.
That is, as a medical profession, you are showing concern. When it comes to the bare
As
situation,
said,
yes.
It is up to the judiciary, or they may count your.
It helps the defense person. That's why the prosecution doesn't want to put multiple statements. You are producing one statement.
Right.
Tomorrow the victim is sent to FSL for collection of blood samples. There the doctor will start recording another statement. Even asking whether the person suffers from genetic abnormality also, they say it is infringement of privacy right.
So that's why I put this question to you, because you are advocating.
Yes, I have seen both sides of it. We used to record, are you suffering from any genetic disorder, your father has any genetic order, we used to say yes, no, even now they write yes, no. But when it comes to say, we say, sir, see, they went into the depth of the family history to influence the prosecution to produce this report. Then the judge gets wild. Then he fires the expert. Khatam baat.
Dr. Rao, but through my experience, I have written history in almost every cases, and in most of the cases, it was relied and accepted by the court. Right? What we ask, what happened with you? What for you have come for examination? So we write all these things in a chronological order, and in opinion, we also mention that the findings are consistent with the history, not with the statement. History, what she narrated.
That says the point, sir. There is a word. Yes. See, what is law all about? Interpretation of statute or law.
Yeah.
You are saying statement, I am not saying. She stated. There is a difference. Statement is covered under Indian Evidence Act. Right? No, please bear with me.
Okay.
Going very, as an expert, or I am telling you as an expert, you are saying she, as per her statement, not to be recorded.
Not statement, it consistent with the history.
That is allowed. That is allowed.
But these people are saying that.
You shouldn't.
These people are saying that when you record a history, it becomes a statement.
Yes.
So that's what I wanted to clarify.
Yeah. You are going by requisition alone.
As sir has said that many times we write this opinion that the findings are consistent with the history given.
And whether history should be considered a statement or not depends on the court.
No, anything recorded is statement.
Yes, it will.
No, sir, court will not. No, court will not decide. Court, sir, uske liye anda kanun. It will hear, not see. It will hear prosecution, it will hear defense.
Yes.
Whichever language pleases it, it will accept that. So you go and say, I have recorded history, it is not a statement. Prosecution will say anything stated by the victim is a statement. Under Indian Evidence Act is a statement.
The decision will be taken by the
We'll
court.
move little further, sir. We are eating into time of Moinek. Otherwise, he'll take me outside and bash me. Yeah, we shared a lot of good years together as postdocs. He was a crucial witness who went missing. CBI was searching for him. He was not found. Ultimately, in outskirts of Uttar Pradesh near Gorakhpur, they found a body. And CBI decided, yes, this body belongs to that Ram Kumar, who is the mediator. They sent the skull to us, then we said it doesn't match. Then they said no, because government is there, PV Narasimha Rao influenced the report, we will send it to London. There was a lab called FSS, Forensic Science Services, which got closed down. So they said, yes, we opine with the CDFD report, therefore this person's skull doesn't belong to that person, that is one thing. Tandoori murder case I have already discussed. Mattu case was the only case where it was overturned at the trial stage. DNA evidence was set aside. It was set aside and it went up to Supreme Court, and here Sushil Shah, this person was Sunil Kumar or somebody, Santosh Kumar. Santosh Kumar was convicted, is still in jail I suppose, right? He is still in custody. This was a very...
Why in this case it was overturned?
Because and I went to repair the evidence. He was little skeptical about DNA testing. He said, no, how can it? It was all about the quantity of blood collected by a doctor and the quantity of blood received by us. There was a discrepancy of 1 ml. Actually, what happened is those days we never used to get those airtight things. You know, used to be the injection bottle, which even now we can use if it's properly sterilized. The rubber cap would be put and a tape put. So the blood spilled out during transportation. So because of that, he said that blood was taken and put on the private parts of the victim to show it as semen. Now, how we tried our best to explain to him that, sir, that doesn't contain seminal parts and it's not semen, it is not having. He didn't believe, but ultimately it was upheld by Supreme Court. This is crucial. Guidelines have to be created for uniform testing. Medical legal professionals need to be deputed to DNA labs for programs under CME is one thing which you all have to consider. So that they get hands-on experience, they can be better tuned to collection. All forensic labs have to be made independent of state government control, police department. Autonomicity is very important because now after all the money is spent, I was talking to you in the breakfast time, is that the money is allocated to FSL. And in Andhra, it's pretty bad. Even after the 3-year-old Innovas are changed, money remains, then the money comes to forensic lab. That's the present status. DNA training education for police, forensic scientists, judges have to be held periodically for refreshment. Proficiency testing is what missing, but these people are themselves subjecting them, but it had to be mandatory. Governments have to provide grants and fresh national accreditation standards should be established. So this is dedicated to my sir. So it will be the best tribute we can make to this great man is when DNA reports would not be set aside as being unscientific. That will keep his soul always rest in peace. Thank you.
Now, I would like to invite Dr. Moinik Banerjee, sir, scientist in Human Molecular Genetics Laboratory, Rajiv Gandhi Center for Biotechnology, Trivandrum. He will be discussing about the technique of DNA sampling in brief. He is also the Vice Chairman of ADNAT, that is Association of DNA Fingerprinting and Associated Technologies, and he established the DNA fingerprinting facility in Kerala. So I invite you, sir.
This particular CME for clinicians, and I am very happy to meet a lot of my old friends here. And the issue is whatever topic was given to me, I think it's already been covered by many experts here. So and many of them will be repeating of what people have presented. So I'll quickly skip some of those slides, and I did not mind giving more time to GV also further, because most often, whether you or me or any one of these people actually face a lot of issues only going to the court, not doing the work here in the lab there. So therefore, spending more time with GV would have been much more better off, because he's a deadly combination, not only of molecular biology and also the legal aspects there. So the life had been so easy for us for so long. We had been able to fool the judges. We had been able to fool the lawyers. Now we are not going to now we are not going to be spared off. But GV, you have to spare everyone. Otherwise, the science will not come to take care of the people there.
That is
Yeah.
set in some kind of.
Yeah. So therefore, your role becomes very crucial. That you should actually try to accommodate science into the translating correct science there. And at the same time, one should not really get into the DNA testing all of a sudden, unless you have certain things in place, like record keeping, very precise record keeping, then your history taking there. Now the history taking is actually a debatable issue that was discussed earlier. And particularly when, and history taking becomes very important, because the history that you get from the police personnel is actually not an adequate history taking there. So therefore, to safeguard your own interest, it's better that you should take a history, but possibly don't include while submitting the report. When you are cross-examined during that time, possibly you can think of including those history taking there. But do it in such a way that
it looks very transparent there. It's not that you have done in a hurried way in the last moment just to avoid the questioning there. So, in fact, I was wondering, most of the people have talked about DNA testing methods and all. And I think one of the major issues that people have been discussing is about what is the best material, what is the best testing protocol. And I'll show you what is the best testing protocol should not actually be determined by people who don't know the science of DNA testing or don't know the population genetic aspects at all there. So I'll show you why population genetic aspects become extremely important. And one of the biggest drawback of history of DNA fingerprinting, even in the country, what GV was telling, what the person next to him, who subsequently took up the case, and he blundered the entire issue. And one of the reasons why he blundered the entire issue is because, first of all, he had no background of DNA testing prior to this particular activity, right? And therefore, and he hurriedly took up for the political reasons to do the DNA testing, and they were not mature enough to present the case in the court, and as a result, the entire blunder happened there. So whenever you are actually disposing of, it's better that don't be very reactive, because we cannot afford to be reactive there in the court there. Lalji Singh can be afford to be reactive because he is considered the father of the DNA fingerprinting there. So, so therefore, we all worked with him. He's very, very short-tempered. He doesn't care about the Prime Minister also, and he doesn't care about the Home Minister, or he doesn't care about anyone there. And I had a couple of examples with him on a personal note, that once I was physically manhandled by a police in Guwahati, and then and my Walkman, my this camera, and some of my samples were thrown out from the window there. And then when I told him this story, and we were staying in the same room there, so when I told him, he immediately said, go and get the telephone records of some of the IB officials there. So I know that once he gets into action, he's not a man of just words there. He actually acts really there. And I know that this particular situation is going to be even worse there. And believe it or not, we stayed there for almost 3 days. On the third day, the DGP, it was there in news throughout when I was in Guwahati, that such thing happened in Assam. And then on the third day, the DGP of the state actually hosted a dinner, and we were all drinking and discussing it. The DGP comes to me and he says that, see, Dr. Banerjee, this is a very common issue in the border state. You are not aware whether it was a BSF, whether it was a state police, or whether it was CRPF. So we don't know who manhandled you, and therefore it's very difficult there. Lalji Singh all of a sudden came and he said that all people were drinking there. So he said that he's not talking to Tom, Dick and Harry, he's talking to a DGP of the state. So mind it that an answer like this from a DGP of a state is not really good there. He forgot about all his drinks and all. Then he said, don't worry, we'll try to resolve it. And I came back to Hyderabad in 2 days' time, I got everything back there. So if the police wants, they can actually resolve the issue like anything there.
They actually found my missing spectacles in a tray
Right.
when I went to the Premananda case.
Yeah.
Believe it, you don't find luggage, but they could recover my spectacles. The police wants, they can get anything.
Right.
That was not costly, I think.
So the power of
that was left in the train.
So power of Lalji was much more.
Recover if they want.
So what I wanted to say is that power of Lalji is much more than a DNA technology bill. So this is, I think I'll skip it out, what is forensic science and all that, which simply means that if anyone has a complaint, he can file a case, and it's the job of the investigator to resolve this complaint. If the complaint needs to be resolved by biological evidences, then one may go for a protein or DNA based evidences. And proteins and DNA based evidences are all about patterns there. And these patterns are molecular signatures, and I'll show you what are the discrepancy between the protein patterns and the DNA patterns there. So therefore, which he tried several times, it never worked. But this technician actually tried at least 2 to 3 times, and it worked very effectively there. But still, the acknowledgement is actually in the name of Ed Southern there. course, as GV again said that Lalji Singh developed this probe on 1986, and but like we have a scientist like Watson, without doing anything, actually he is credited for his Nobel Prize. Similarly, we have another scientist who also grew around the same time when Lalji came up. He was very smart enough. He also saw that there was a specific sequence pattern in Lalji's sequence there. What he did is he actually synthesized those set of sequences there and then made it as a probe there. In Lalji's this BKM probe, there was a GATA repeat sequence element there, and which used to hybridize with and give you a multi-locus fingerprint profile there. And what Georg Kaplan did, he actually synthesized those GATA repeats and he used the synthetic probe, synthetic sequence as a probe, and then he also was able to generate similar fingerprint patterns there. So therefore, being smartness actually helps you out to win the race quite often there. And Georg Kaplan was one of those there. And so fingerprint pattern, we know that it is all about sharing of genes or alleles from your father and the mother. And therefore, you will carry 50% of the alleles from your father and 50% of the alleles from your mother there.
Very artistic, very good.
Very unique.
Right, thank you. So, and these are the various sample evidences that you can actually get. One is blood, semen, saliva, urine, hair, teeth, bone, tissues, anything biological, anything that has a cell, living cell or dead cell is of evidence there. But although the investigators will say that it is possible to do fingerprinting with a single cell there, it is possible definitely there. But is it really practically applicable and has it been validated by various laboratories? It has not been validated by various laboratories there. So therefore, don't get into the game plan that even if GV questions that you could have done it with a single cell, why you could not do it, right? So it is not really practically feasible there because it has not been validated extensively in the field of that. It's published. So
you don't have the expertise.
So, yeah, so that is where the expertise will be questioned there. So typically, to generate a fingerprint profile, it says that close to 200 to 500 cells we really require to do to generate a very precise fingerprint pattern there, and the number of the amount of DNA that we'll get it from each cell would be one sperm, we will actually get close to around 3 picogram of DNA. From one cell, you will get close to around 6 picogram of DNA. From one hair cell, you will get approximately 1 nanogram of DNA. And from one hair plug, you will actually get close to 130 nanogram. This is all estimate. Now the thing is, this is on our absolute values there. You will never get such things. So and I will not ask GV to see this particular slide there.
We worked on it, so we never got it.
It is next to impossible that you will actually get these quantities there.
Given in Butler book.
Mm-hmm. And
1 nanogram per hair.
And one drop of blood will actually give you close to 1500 nanogram of DNA there. So what I do is whenever anyone comes for training in my lab, I said that the first thing that I will ask them to do the DNA isolation, and I tell that I can evaluate a sincerity of DNA isolation by looking into the color of the DNA there. So therefore, the color of the DNA, if it is completely white, I will say yes, you have done very sincerely there. The moment the color of the DNA keeps on changing, so that will show the sincerity there. So therefore, I will say that if color is not completely white, then I will say that the particular person has not done the DNA isolation very sincerely there. So earlier days of serotyping, we had actually blood grouping methods. Then we had, actually this is all carried over, right? So then this is all, you all are aware of it, the blood grouping and all. And like yesterday, Pankaj was also saying that if I ask how many of you are actually O blood group, I think 90% of the people will turn out to be O blood group there. So therefore, there is no point in really asking about what is the blood group there, based on serological typing. But now the catch comes. Based on the blood grouping, you have only 3 groups there, or 4 groups there. But I can still do the blood grouping and still very clearly determine are you really O group and to what extent you are O group there. And how? By doing this there. So although at the protein level we have just 3 groups, but at the DNA level you can see that ABO we have, I'll show you in fact in the next slide how many alleles are there. So there are 29 known blood groups which are determined by 200 antigens there. By sequencing the 31 genes that determines the surface antigens, hematologists can classify the blood group into 29 groups. A person's full blood type might be read as AB, D+, M+, or something of that sort. A single glucosyl transferase gene on chromosome 9 controls the ABO antigen, and the version inherited from each parent and person determine and his blood grouping there. And therefore there are close to 100 alleles for ABO blood type there and more than 200 for the rhesus system there. So if you go by DNA based evidences, even for blood grouping, you can very precisely tell about his blood group there. But serological based typing, because the protein is actually very prone to degradation and all. So even if you're not able to do that, you can actually think of doing it using DNA based typing there. But it's still not a very mature system to do it there. So, and there is still no data available to very precisely determine which of these alleles really very clearly determine the allelic patterns there. And what I mean by saying so, I'll show you in another slide there. This also I think people have talked about. Is about, so another serological based typing is HLA typing there. And one of the most conventional basis of HLA typing which is extensively used is HLA B27 for ankylosing spondylitis there, right? And if you look at the serological typing, it's all HLA B27 there. But when you look for HLA, DNA based typing for HLA B27 alone, you will have close to around 29 such DNA variants there for HLA B27 alone. And luckily, I came across such one of those cases. So one of my students actually turned out to be HLA B27 positive serologically there, and she was actually working on population genetics of HLA genotypes there. And so whenever you are found to be serologically positive, you presume that you are likely to have any of this ankylosing spondylitis or group of those particular disorders there. And so based on that, whenever she will go for any flight and all, she will possibly carry weight, and then she will say, she will have psychological pain there. Which I still say it's a psychological pain, and based on that she went for various radiological imaging and all. She was found to be completely normal there. And so then I said that it's better that you go for a extensive screening of HLA-B27, and then she went for extensive HLA-B27. She did not have any of these variants which actually which is responsible for ankylosing spondylitis. Among these 29 variants, these are the only variants, 7 variants, which actually are responsible for ankylosing spondylitis at the DNA level there. So this is what I mean actually the difference between serological typing and DNA based typing there. So therefore, you can actually replace those serological based typing with the DNA based typing today, whether it's for blood grouping or whether it's for HLA typing there. And these are the so many alleles that you can actually see, close to around 10,000 alleles that you have for HLA itself there. This is for HLA class 1 alleles. At the DNA base, we have close to 10,574 alleles. For HLA class 2, we have close to 3,658 alleles. This is all DNA based alleles there. So total, we have close to 14,232 alleles there. So therefore, there's a lot of scope that you can still fish it out based on DNA based evidences for HLA alleles there. So this I'll skip. I think this has already been covered previously. Collections, specimen storage, extraction. Specimen storage is definitely extremely important, that even if you are actually thinking of, although you are a forensic division, whether you are getting into DNA testing or not, but you should still actually have a very good repository or biorepository where you can actually keep all the samples whenever it comes to you there. So you should definitely have a -80 or at least a liquid nitrogen storage containers there. With a small liquid nitrogen plant, so that you can actually replenish the liquid nitrogen as and when it gets over there. because quite often these samples, because being a forensic medicine department, you are supposed to maintain the samples also there. Unlike for many of us, like I don't have to maintain the samples, I can actually really get rid of the samples the moment it comes to me. Once the report is done, I can actually straight away send it back to them. So this is also, everyone is aware that yes, DNA fingerprinting is, this is how the DNA fingerprinting is done, that the child inherits one particular band from the father and one particular band from the mother there. This is a routine fingerprint pattern there. And now the issue is that this child can inherit this particular band from the father and this particular band from the mother. Child can inherit this particular band from the father and this particular band from the mother also there. So as long as you get this type of pattern, it's extremely safe there. But the moment you get this type of pattern, where the father, you inherit this particular allele from the father and this particular allele from the mother, you are likely to be homozygous there. And this is the real issue with the Indian scenarios there. We are actually in extensively inbred community there. We keep on getting married within our own communities and all that. And as a result, most of this heterozygous alleles turns out to be homozygous there because of extensive inbreeding and all that. So therefore, we are in India, we are likely to become more and more of homozygous alleles there. And that is where the population genetic data becomes extremely important for the DNA evidences there. And this with my personal experience when we were actually doing DNA typing using those mini satellite markers. So what we did is we were screening the tribal communities there. And in a southern blotting of 12 samples, I found 7 of them actually had exactly same single locus mini satellite profiles there. Now what happened? What does it mean? It means that if one person commits a crime and other person is caught, both are actually put behind bars there, or the wrong person is put behind bars there. But this is important. How do you literally figure it out that that the sample that you have is actually unique from the global point of view there? So we actually use this formula very extensively used as P is equal to 2 into P and Q there. So P is one allele and the Q is another allele there. And what we do is we try to find out whether the sample that I have, what is the alleles that I get it there, like this. Plus has allele 18 and allele 16 and allele 17 there. What I do is I try to find out what is the frequency of this particular allele that has been reported in the population till date there. And what is the frequency of this allele that has been found in the population in this database till date there. And based on this particular allele locus itself, I know that the possibility of finding out this fingerprint profile is likely to be 1 in 10 individuals there. 1 in 10 or 10,020. Anyway, 1 in 10, 1 in 1,020, right? So anyway, so this is how it goes on. For example, just showing here, allele 16 and 17, this is the allele frequencies for this particular locus, and therefore I put this formula. Now using this particular formula, it is 1 in 10 individuals there. So using one particular locus, I can find that this particular person is unique only among 10 individuals there. And then I keep on adding the number of locus. I use it, and then I use the same set of formula, and then keep on adding P1 into P2 into P3 and P4, and whether it's 23, 26 or 29 locus, and then I can actually increase my my level of confidence of my data. And using this set of 13 Cody's marker, one can actually find out that it is unique in 1 in 1.56 quadrillion population there. Now I think that's why the quadrillion population there. So it is 1 in 1.56 quadrillion there. But when you go to the court of law, they will say then, so, but what happened to that 1 in 1.57th individual there? But the issue is, yeah, so the issue is that, what is the world population there? The world population only over 7 billion there. So do I literally have to go for such a level of authenticity there? It is not really required there. So therefore then the question comes is that when my world population is only is only around 7, or even if I go for 10 billion itself there, let us to be in the safer side there. So I should be able to discriminate my identity only in 1 in 10 billion individuals there. That should be sufficient enough to actually say that yes, my test is very, very powerful there. And doing so, I can actually reduce the number of markers, and by doing so, I can actually bring the cost factor for the testing very, much lesser, and possibly it will be much more clear, right?
Yeah. Even to say 1 in 1 billion is sufficient probability, man.
Yeah. So now GB says, you note his word there, huh? He says 1 in 1 billion is sufficient enough there, but in quote, he's not going to leave you there.
No. Frankly speaking, these numbers of quadrillion and all creates a doubt in the mind of the court. He'll say, have you ever tested this many people to say 1 in what? 10 billion, 30 billion, when world population is only 7 billion.
Right.
So realistic presentation is very good.
So to convince what he said is actually he's right that I don't really need to worry about 1 in I need to worry about 1 in 1 billion. I'll say no, I will not even look for that one there. Say for example, the Malayali speaking population, if I think that it is around how many 50 million, let us, so that is where he has a point that yes, I don't really need to talk about 1 in 100 billion or 200 billion or quadrillion there. I should be able to tell that what is the uniqueness of a particular gene pool there, and bring down the cost factor significantly down there. And this is possible only in India there, not in US or European setup there, where there is a mixture of population. There is hardly, they all keep on interbreeding among each other there, and at the same time there is a lot of children are being coming out through the IVF and all. So therefore you have absolutely no idea that whose kids are they. And to justify what he said further is, this is a very good observation that I can really identify the uniqueness of an individual based on allele sharing patterns of a locus, and then I can find out what is the pattern of 100 locus and their allele sharing patterns. I can then color code them separately, and I can say that I can identify at a global level all the African gene pools there. Here I can identify all the Europeans here. I can identify all the East Asians or Mongolites here there. So therefore, I need to really identify the uniqueness of a Mongolite gene pool only in this set of, using a set of markers there, which we now call as ancestry markers there. So using ancestry markers, we can actually really bring down that. And ancestry markers are relatively cost effective there. And then now this is this is the world population. This is the South Asian populations, and this South Asian populations I can still distinguish based on the same set of allele markers, and using another set of statistical parameters, I can actually distinguish my Indian population into 5 different gene pool status there: Indo-European, Dravidians, Austrasian. And now what I can do is I can further stratify this Dravidian community in my Kerala gene pool, that they actually cluster as 2 distinct communities. One is the non-tribal community, one is the tribal community, this is the non-tribal communities there. Right? So this is called stratification issues there. So using the stratification issues, actually I can really bring down the cost factor of DNA testing much lesser there. And this is where the population data becomes extremely important. This I'll skip it because I think many of you have shown earlier. And this also I'll skip it because again it shows that yes, this is the father and mother's profile, this is how the kids are going to be. But it is not so simple there. I don't know whether anyone has shown this problem slides, but GV, I suggest you close your eyes, although you're aware of it. So this is what actually the real issues of fingerprint comes there. I think you all should be aware of it, but don't get nervous about it there. That is where the population data becomes very important there. And therefore I have to literally bring out the database frequencies and the mutation frequencies for the D7S820 there. And based on that, I can be very confident that yes, this is a mutation there. And therefore, to convince further, so now that is where the 13 codes will possibly have to go expanded to 19 codes to 29 codes there, right? Or possibly then I have to, and if you're still not satisfied, I have to add HLA typing, I have to add DNA based blood group typing, and then convince it with, so that's where the increase of markers are important there. So when you come across such cases, you will have to address the issue of addressing more with more number of markers there. So this is one locus there. Now I think the semilogenin marker has been also been discussed extensively. I think the peaks are not visible there. Again, here you have father's allele, father's X allele you have seen, mother's X allele you can see, daughter's X allele you can see. So ideally one would say that if he can question me in the court of law that this sample is completely screwed up, where is the father's Y allele there, right? So that I have to literally convince him that father's Y allele is actually mutated there. Then he will ask me what is the evidence that you can say that the father's Y allele is mutated there.
Yourself are framing questions, you are answering.
Yeah.
Give me a chance.
So then I have to convince him.
Now with the technology we have answer for both these.
Yeah. So
we have a different kit which is altogether giving you profile on 23 different markers. That is paternity 23 complex paternity kit. That is having one marker on all different chromosomes, all non-code is markers.
Yeah.
And for this issue we have double filar and fusion 6C which are having with this amylogenin one endal marker, one 3 or 4 biaxial markers. So both these questions can be handled.
Yeah, so it means that he says that keep on collecting as many number of kits that you can, which is and use it whenever you want it. Yes, the issue is that becomes a real expensive issue there, because this type of issues you are going to have it one in. We could get it only one in out of 10,000 out of around 2,500 cases samples that you have screened there. So
Can I just interpret, just add on here one a less technical aspect which just turned out, which I thought needed attention. When we are doing a particular work where we know exactly the questions, the errors, the confidence limit, the how well we can stand by it, come what may, or is there some evidence beyond reasonable doubt or beyond any doubt, is when we are giving court evidence. Another factor which is always dominating is we can see which are the questionable parts. When you are putting question and answering, and he's saying you are already putting the question, you're not allowing, I can ask, I may or may not have asked those. That particular bit is what happens as soon as we see the document in the court. You're screening through before you are going in the witness box, and you know exactly the spots which can be questioned, which should be questioned. If there was a right question, my answer would be wrong here. You have played the whole chess game there.
Right.
That whole chess game where you have seen 16 moves ahead, and then none of those moves happen half the time, or rather 90% of the time. None of those moves even won.
Right.
But you've seen where the opposing lawyer, if he was competent enough to see through your eyes and your competence level, could have made your whole report porous. He could have blasted right through, which is what is the sign of that expertise.
Yeah. So
when you are already getting the questions for all your work.
Yeah.
And
so I can tell with 100% confidence that how many judges, how many advocates are there, lawyers are there in the country? Close to around 10,000.
Every bar association has more than 5,000, 6,000.
Okay, let us assume that there are 1.2 million lawyers are there. I can be very confident there is only 1:1.2 million lawyers there. Probability there, because there is no one else with such an experience there. That's what 1 in 1.
Adding to your point is, it's also what I observed now. We used to prepare, we used to discuss a case before we go to the court, because we are all sensational cases. But nowadays, they don't even come with the office copy of the report to the court experts. They come there, the constable gives them the court record, he turns the bundle, and there he sits and reads and he says, ah, fine, I'll now enter the box. That is where reports are failing. If the preparation, as you say, put yourself in the accused place and see what questions can come, I think conviction rate can increase based on scientific evidence.
But sir, we are not able to get which case we are called for. We never know that.
No, but they will mention your case details. No.
In the summon, we never get the case.
No, but you can ask for it. You can ask for it.
How?
Yeah. Whoever is delivering the summon, you can ask that I want the case report there.
He has no relation.
No, but the court is supposed to provide you that.
This is what we do actually.
Yeah.
Actually, we get just 2 days before, or maybe 1 day before.
No, sir, you can go to court and also seek time for, because you can say a very limited information.
Almost 100 case. I'm telling you, 100% case. We never know which actually case we are called for.
No.
But we used to maintain a register where crime numbers year-wise, whatever cases we carry a FIR number or a crime number, right? So crime number, CDFD number, and the date of dispatch or date of the report. So when they send us the summons, we tally it with the crime number. It's just control F, control search. That's it.
Basically, sir, most of the time we get we do the postmortem of a victim,
Yes,
right?
yes.
And that someone comes from in the name of an accused. Number one.
Crime number will be the same.
Crime number is registered after we deposit our report. Because
FIR number will be there.
FIR, but FIR is usually not there.
No.
CC number comes to the
No, but you can definitely ask for the one of the condition that you can do. I don't know. We can afford to do saying that I don't issue me a summon to appear in the very next day there. Give me at least 15 days time there. And similar conditions you can also say that I am not supposed to dispense off with the evidence within 2 days time. You have to give me. You can actually put it in the transparently there. In the website that I need 15 days time to appear in the court there.
We put it in the covering letter.
Yeah.
We started that trend there.
So you can say that, and then they are bound to follow the rules there.
Sorry to say that,
Always mentioned in your report, then when calling for someone so and so should be called with giving the
No.
reference
So
number.
we attach the terms and conditions of issuing a summon to us there.
Sorry to say that now we have started receiving someone on WhatsApp, sir.
That is now allowed. That's allowed.
Means the same day.
Yeah, but that is simple. You can say that what you do is
Insufficient time, court will allow the experts.
Yeah. You can respond it through your office saying that I need a minimum amount of time there.
MP state.
Anyway, so this is the issue, and so we confirm that the Y chromosome was intact, but there was a mutation in the amylogenin Y itself there. So therefore, this is actually the same male, so the mutation was confirmed there. It's been taken from, if the individual has undergone transfusion in the last 1 month, you should actually record it that there was no blood transfusion that was taken in the last 3 months there, to be safer side there. So such issues you really need to record it there. So this is an issue of an oral cancer patient there, taken from the blood of an individual's DNA profile and the same individual's oral cancer tissue there. You can see that the DNA profile of the blood sample and the tissue sample of the same individual doesn't match there. There will be one allele that will be matching, but the other allele there is a shift there. Yeah. So you can see that this is the same individual. You can see this is one allele, this is another allele. These are stutter bands, and this is of the same individual's oral cancer tissue. This is one allele which is matching, and this allele is actually a shift there. So you have series of such issues in the various cancer tissues there. So therefore, this is called microsatellite instabilities there. So therefore, it is important that you really take it for your own safeguarding principles, that you should actually take in history taking. And if they don't agree, you can quote such issues that yes, it's important that history taking should be done there.
Sir, what if the organ which we are looking for is a transplanted organ?
Oh, yes, that is also an issue. So you should also note that one also, that whether it's a transplanted organ or not.
Transfusion is also a part of
Whether we will get the DNA profile of the
donor or you will not get of the donor there. Yeah, you will get actually mixed profile.
Recipient and donor DNA.
This declaration is already there in identification form that no transfusion was done, no organ transplantation was
Not
done.
only transfusion, transplantation was also not.
But if the case is
But if the case is like unknown or dead body, yeah, when we are doing cleaning.
Yeah, that
you have to, then you have to retest with other organs. But it will be very clear double profile.
Yeah. So it's, if you literally get such a profile, then you can actually take it out from the blood samples or any other body tissues or any other organ tissues, because if it's a dead body, you can actually take any body tissues anytime, right? Yeah. Said that's called microsatellite instabilities, and I think this also I'll skip it because many of you have already spoken, right? So therefore, this also needs to be noted down if you actually come across such issues there. But don't report exactly like this in the court of law, because you will be screwed up like anything there. Gears there. The gears is, so far it is the DNA. We do the DNA testing only when it is sent to us, and therefore I cannot be involved in predictive testing there. So that I just have to do the DNA profiling and then try to see whether the database has similar matching or not. Country is actually moving towards that particular direction, and I think another insignificant, very significant person, but an insignificant knowledge on the DNA based evidences is actually going to head a program of 10,000 crores on the human genome mapping program there. So therefore a lot of thing is going to come in the public domain, and many of the questions I think that was asked is that, is this the human genome sequences or the DNA profile, is it of do I have the privacy rights and all? I actually don't worry about it there. And because if you do a search, you will get almost 1 million DNA profiles of more than million individuals available in the public domain there. Today I cannot publish anything on the whole genome sequence, on whole DNA expression profile, or whole gene expression profile, whole methylome profile, without uploading my entire data in the public domain there. So therefore, there are more than millions of such whole genome profile, whole gene expression profile, whole microRNA profile, whole methylome profile is already uploaded in the public domain, which is available free of cost, and with individuals' identity also there. About our horoscopes and all, right? So I will say that be very careful about horoscopes. So this is the horoscope of a child who was doing his second year of engineering, and he had 2 elder sisters who were actually waiting to get married there. And so whenever the daughters are likely to get married, or the parents get excited about matching the horoscopes, and therefore this were the parents who were educated parents, and so this is the mother who writes a letter to me saying that while going through a book of horoscope, my husband discovers that appearance of moon and Saturn in a single box of horoscope of a child confirms that the child is the son or daughter of a person other than mother's husband. So she gave me the photocopy of the book also there, and the same is the case with my child, and this has created doubt in my husband's mind there, right? So you are going to get such cases also there.
No, we already got. Yeah, this
call there. I said nothing doing, because tomorrow if the evidence goes against you, you're not going to leave me there. So never get into such traps, because DNA evidences are such that tomorrow if you literally come out with an evidences which actually goes against the person who is actually come to you, you always encourage him to come through the Women's Commission or through the court of law. Otherwise you are going to be in a big soup there. And then and this was the case from Rajasthan which came to us there. So and then I tried to convince him that you take my name to the Women's Commission of Kerala, and then he lodged a complaint, and then the Women's Commission encouraged him to come, and then the case was sent to us, and this was a very challenging issue. If this turns out to be in favor of that particular guy, I thought I'll leave genetics and I'll start worrying about astrology thoroughly. Fortunately, the genetics prevailed, and the horoscope turned out to be wrong there. The horoscope interpretation is wrong there. I will advise you literally, if you literally want to go using want to use this horoscope, I would suggest, yes, don't believe using this particular method, horoscope based method. If you want to literally do a matchmaking, I would suggest there is a very good paper that was published in Nature Genetics a couple of years back in 2002 or so, which says that one of the best matching protocol that you can actually think of is HLA mismatches there. So your husband and wife should actually have a mismatch of HLAs there. So then you are going to have a better match probabilities of compatibility. So that is one domain which possibly you will also come across. Then I'll keep on shifting to some other domains, and how do we interpret such domains then? Now I'll shift gears towards how we can actually talk about predictive testing and all, and how can I literally find out the motive behind the killing also there. So let us assume that these are three individuals everyone knows very well, and this is based on SNP typing there. So what you have, most of the sequence is same. You have one particular variant here, you have another particular variant here there. I think the bar got shifted. So these are three individuals, you all know these three individuals there, very famous persons there. So this is George Bush. So George Bush and Bin Laden are actually quite similar there, right? At this particular point there. So therefore they both are having AA there. Then what I do is I try to identify what is the similarity between these two. So what is the similarity? Don't worry about the color there. Let us understand from behavior point of view there. So if he wants to destroy Iran or Iraq, he will do that. And if he wants to destroy America, he will do that. No one can stop him there. So I will say that this particular A stands for being adamant there. And this T, so therefore if he is people's choice, so it means that he is adamant, working for a positive cause. He is peace loving, working for a positive cause. He is adamant, working for a destructive cause there, right? So this is how the change is likely to happen in due course of time. And is it really true? You may ask, is it really true? You are not going to understand unless and until it actually goes to the court there. My publication series of publications of people will keep on publishing hundreds of articles. No one is going to believe it. Unless it goes to the court of law, you are not going to accept it there. So this particular tempered, I know that there is a genetic distinction between this particular group there. So there is a short-temperedness and the normal tempered individual there. So short-tempered doesn't have to be pushed towards the wall to react there. He will react instantly if he is not happy with it. But a normal tempered individual, he has to be pushed to wall, but he also has to survive there. Now when he is pushed to wall, he will actually try out all defensive mechanisms, and therefore he convinced the jury that he killed the person only as a defensive mechanism to survive. Otherwise, he did not have any motive to kill him there. And therefore, jury did not knew what to do. They were thoroughly convinced with his evidences, and therefore they reduced his sentence from 9 years to 8 years. Anyway, he has murdered someone there. So such issues are also likely to come there. And another issue that is going to come up quite often is about like drug abuse or drug issues and all that. So this person died in medical college, Trivandrum, and whenever a person dies, there is an investigation, and an investigation team established that he died because of overdosage of carbamazepine there. So and therefore the doctor was harassed for this particular incident. But if you literally ask me, is it really true that he died because of carbamazepine overdosage? I'll say no. Because individuals having such genetic makeup are more likely to have side effects of the carbamazepine there, like Steven-Johnson syndrome there. So such issues are also likely to come up to you. So and there are so many cases like this. This happened again in Bhopal in itself. So this person died, and this person was actually put in a hospital there, not in AIIMS, but another tertiary care hospital here. And she was diagnosed to have migraine there, and she was discharged in 2 days' time. And later on, in another 2 days' time, she comes out in a completely vegetative state because she had an aneurysm which ruptured. Aneurysm is this one, which ruptured, and she collapsed there. And therefore, she went to the court, and she was actually given a compensation for that. So such issues will also come to you. And another set of issues is this: there are large number of cases of cardiovascular malformations, cleft lip and all. They are of genetic origin, it is believed, but they can also be drug induced there. So we have close to around 250 such malformed cases which are drug induced because of anti-epileptic drugs the mother was taking when she was carrying the child there. So and so therefore there are issues, in particular in Kerala. People are actually quite they take you pull you to court for each and every small issues there. So therefore they come to they come and they say that I want to confirm whether my cleft lip, is it because of genetic origin or is it because of drug induced issues there. If it is for drug induced, then he will say I'll take the doctor to the court there. So such issues are also coming in Kerala courts and all that. And then comes the issue is about predictive testing of an individual's age there. Can I literally talk about the individual's age there? So it is really very difficult to tell very precisely about individual's age. But yes, now the things are shifting. If you literally Google search, you will see plenty of companies in the US which is actually running their own private companies, telling that they can actually tell you about your age very precisely by the telomeric shortening of the of it. So.
Survival age or the age at a particular moment. Or you're talking about how long you will survive.
No. This is your age there.
Okay, at a particular time.
At that particular time. So this telomeric age, actually, as you keep on increase in age, the telomeric shortening keeps on happening there. So one of the best example of that one is the Dolly. You are aware about Dolly, the sheep which was cloned. So Dolly died not because of failure of the test of the protocol. The Dolly died because she had the age of her mother's age when she was born there.
She was old when she was born.
Yeah, so she had the mother's age there. That's what I'm saying. So therefore she died because of these issues there. So most possible answer would be that I am going to commit a crime only after 3 o'clock there, not before 3 o'clock there. So it means that I am not a morning type person there. I am more of an evening type person there. So such testing protocol, if you can really include. Now if I ask you how many of you are morning type, evening type, based on the questionnaire, it can be biased there. I gave the same questionnaire to a couple of my students and I said that you get the same questionnaires filled up from your parents there. So one of the students got it filled up, and so she said that yes, my parents are morning type and I'm evening type then. I said, you give your mobile to me tonight and you go home. And then let me see whether you're morning type, evening type there. So, but the questionnaires, when she filled up the questionnaire, her questionnaires answers were all evening type there. So when you, therefore, one can be very subjective about such issues there. But when I said that, okay, now you are supposed to do the DNA testing of your parents and yourself there, and let me see whether you are morning type or evening type there. So she was actually found to be morning type based on DNA evidences there. And how do we do it? Like this there. There is a particular gene called period 3 gene, which determines your chronotype. And if you have this allele 5 and 5, you are more likely to commit a crime only in the evening time. You are more likely, and if you have 4 and 4, you are more likely to commit a crime only in the morning hours there. So these are all supporting evidences which you can say that yes, this will be helpful there. And then like G was telling that yes, we reconstructed the entire skull architecture. Yes, in fact I was asked to give a talk in craniofacial anomalies conference in Trivandrum Medical College. So so the person who invited me, she actually did her PhD on beauty there. So I realized that beauty possibly lies in the eyes of the beholders there. But when I went into the literature, I said that the beauty actually doesn't lie in the eyes of the beholder. It actually is more of genetic in nature there. And then I went on to see whether is it really genetic in nature or not. And then you realize that yes, there are certain chromosomes and the genetic loci in these chromosomes are more likely to have a different architecture there. So using those genetic loci, you can actually really identify you can reconstruct the skull architecture of an individual there. Yeah, 3D DNA printing using this DNA profiling there. So there are series of such candidate genes that has been already identified for for creating a 3D architecture of the human brain there. So therefore the stem cell therapies, how good they are, how safe they are, and how well regulated they are, that really needs to be addressed there. And this is one example there. When we keep on, when any person has to undergo stem cell therapies, they will keep on concentrating the cells there. And then once you have sufficient number of cells, only then you will try to transplant that cells there. Now what happens, that when you have, this is the first passage, 10th passage, 50th passage of human embryonic stem cells, human induced pluripotent stem cells there. So in the very first passage itself, you have trisomies and monosomies there. So when you use these cell populations to be transplanted, you are not doing good to the patients, because you are actually injecting a lot of trisomies and monosomies in the patient's brain there, if it's an autistic brain there. Or similarly, for depending on what is the nature of the problem, wherever you're transplanting it, not the stem cells there, it can create much dangerous issues there. And it is known that certain chromosomes are more prone to increase in number, like chromosome 1, chromosome 3, chromosome 8, chromosome 12, chromosome 17 are more likely to have trisomies and all that.
So we thank Dr. Moinik Banerjee for an excellent presentation on starting from the history of evolvement of DNA to the various use in the future or in the ongoing research that the DNA can be put use into, starting from the diseases to the criminal background. Thank you, sir. And I think we'll now break for tea, and let it be a short and crisp break so that we can back on time before the lunch to have one more lecture.